We are just about ready to get started in the afternoon. Thanks to those of you who have stuck around, or those who are joining us too. All options are great. As I was promoting FlowText, as I often do to many people who maybe do or don't want to hear about it, this was one of the talks that I was really talking up a lot, because I think talk from Karen about panel design is probably about one of the best that you can possibly get. She's got a ton of experience. She does it with a bunch of people, and I've seen a ton of extremely beautiful data that comes from her help. So uh, I'll let her get started on it. But Karen Clise-Dwyer is a professor in the Department of Hematopoietic Biology and Malignancy at MD Anderson Cancer Center here, where she directs the cytometry and cell sorting facility at South Campus. She earned her PhD in biochemistry and immunology at the University of Wisconsin-Madison, and that's where she started her work in cytometry. Under her leadership, the ACSF has grown from a lab with 4 instruments to 16 major systems, including a robust spectral cytometry service and a new service in single-cell multiomics. She is an author of more than 50 publications and leads a research lab focused on the application of cytometry and cell sorting to the study of leukemia and cancer immunotherapy. And she is one of the co-founders, the scientific director of FlowText, and which is, of course, this Texas-based cytometry society. And she gives a great talk and is a great mentor. So welcome, Karen.
Okay, so this is the first time I'll have given this talk on panel design. So hopefully this goes well. You're all the guinea pigs. Enjoy. Okay, there we go. So just a title side, we know what flow cytometry is. We're looking at a single cell level. We want to be able to use large panels to get big data out of single cells. In this talk, I'm going to mostly talk about immunophenotyping or antibody-based analysis of largely immune cells. However, as Jake told us, flow cytometry can include all kinds of different things. And this panel design principles can be applied to any sort of assay that you're running. My object here is really to tell you about how to think about panel design and the process that I would go through to design a panel, all the way from a simple panel to something more complicated. And I'm not going to work you through how to do this in any kind of software, but more how to think about it. So your goals here is to get the best resolution you can. I would always tell you, if you think you're going to run a big panel and you want to start smaller, you should plan with all the markers you think you're going to need. It's much harder to start with an 8-color panel and make that a 16-color panel than it is to plan a 16-color panel and run half of it, right? You should always focus on commercially available antibodies, because if you start going off into non-commercially available antibodies, your experiments suddenly multiply in orders of magnitude as far as the size of them. And always use the entire configuration of your instrument. Use all the lasers and spread out your panel as much as possible across the emission spectrums for your detectors. Okay, so the step 1 of panel design, regardless of what it is to think about what your cell types and markers that you're interested in are. So I'm giving you a couple of examples here. One is a pretty simple 8-color panel that would look at T, B, and NK lymphocytes, trying to determine just differences in frequency between these, between treatment groups, maybe patients, cancer patients who have had different treatments, or you could do something similar in mouse. And you're going to want to decide what the markers that are relevant to your cells are and know if they're positive or negative on each cell type that you expect to be present in your sample. So you need to, when you're thinking about panel design, you have to remember what cells do express and what they don't express. So you want to be looking at that whole context of markers. Then you can move to sort of a more complex example. So I might want to be looking at T cell function and exhaustion, and looking at not just the frequency, but also the function of those T cells and myeloid cells that I pull out of tumor tissue. So that would give you a much more complicated panel. And there's an example of a 36-color panel here that just gives you a sense of what list of markers might look like. Okay, so protein expression levels is always extremely important to get a sense of as you're planning a panel. There's a lot of data out there on this. It's not extremely well standardized. There isn't a quick and easy lookup that I can give you. But you're going to want to get to a point where you can take your markers that you want to look at and group them by approximate expression level. So there are a couple of papers that I've got on this slide that I can share citations with you guys. But what we're looking for is how many copies of a protein are on the cell surface. So we know that, say, CD45 in any species is going to be very highly expressed, and lots of other markers are going to have much less expression. So there's always going to be much less CD11C, for instance, for DCs on the cell surface in terms of number of molecules than there are of something like CD45. Okay, so next thing you need to know is your instrument configuration. Every instrument is going to have somewhere between 1 and 5. There are some instruments that have up to 9 lasers. These are all at different wavelengths. The lasers can tell you or determine what fluors can be excited. And your common laser lines are a blue laser 488, a violet at 405-ish, a UV at 355, a yellow-green at 561, and a red laser at 640-ish. Most instruments have separated laser intercepts with the stream so that you can use the dyes that have overlapping emission wavelengths as long as the excitation wavelengths are distinct for those dyes. Conventional cytometers, as Joel told you, use one detector for each fluor and have a broader bandpass, whereas spectral cytometers break your light up into more channels, and this allows you to increase the number of parameters that you can use in your analysis by using both more channels and also oversampling your data, which allows you to apply spectral unmixing rather than compensation. So again, use all your lasers, spread out your fluors by emission as much as you can. So when we look at configurations, like I said, you want to know about the lasers that you have, the nanometer range of those, or the nanometer laser line, and the bandpass range for your detectors. I'm showing a couple of examples here where you can see what some of the conventional cytometer detector arrays are. All of your light is separated typically by optical filters, which are just coated glass filters that allow light through at a defined wavelength. Then there's now spectral cytometers. This is the BDA5SE, which uses these modified arrays similar to their octagons. These are cascadagons, I believe they call them. And this is the Aurora, which is a big market player as well, and uses a slightly different detector array using APDs to separate that light out. With Aurora data and with any spectral data, which you can see is the full profile of your spectral of your dye emission. So this is starting at 355 here for the UV laser and coming up to 800 nanometers. And similarly for violet, you're going to start at 425 and you're going to go up to 800. Similar goes for blue. You're going to start just above the laser and go up to about 800, and so on. This particular dye I'm showing you here is APC, and you can see that it's going to give you sub-signal off UV in the far red. It's going to give you a little bit of violet signal, quite a lot of signal off YG, but then most of the signal is going to be at the lower range of the red laser. This is what we expect. Okay, so I like to use a table like this to help me visualize a panel. It's a pretty simple device, but it helps me to be able to see how my markers are arrayed. So this is an instrument in my lab. Well, it's actually 3 instruments in my lab that are 5 laser, 18 color X20s. And these are very good instruments. They have 18 detectors. It's 2 off of UV and then 7 off violet and 2 off of blue and 5 here off YG, 3 off red, okay? And what we can do if we split this up into kind of this matrix style is that we can look at the emission wavelength here on the Y, and we can look at each laser. And if things are close here within one laser, like a BB421 and a BB510, there's going to be more spectral similarity between those dyes. And the same thing goes as we go across. So keep this table in mind because I'm going to come back to this as I illustrate a couple of panels. Okay, so step 3 is choosing your fluors. You need to know your configuration. You should know something about the types and the spectra of fluors that you think you want to apply. You'll need to know about the brightness of those fluors because some fluors are very efficient, and if they are hit with a photon, they'll give off a lower energy photon. Some fluors, it's less efficient, and there will be less signal coming off of those. You should also think about application specific properties like you heard about in the last few talks from this morning, where you want to think about your viability and your intracellular staining. Some fluors are going to be better for that kind of intracellular staining. Like we like small molecules for that if possible. You also need to consider the co-expression of markers and how much you're going to lose signal by co-expression and closely related fluors. Also, I want to focus all the time on just look at commercially available reagents as much as you can. You can do custom conjugates, but you're not going to want to do a lot of custom conjugations. Okay, so fluorochromes. I'm not going to go through all the types of fluors over here, but I'm going to tell you that every fluor, regardless of whether it's an organic molecule like FTSE, a GFP, which is a fluorescent protein, or a tandem fret dye, which I'll tell you a little bit more about on the next slide. Every fluorophore has an excitation profile and an emission profile. So that's the blue for excitation and the red for emission. The difference between the peaks is called the Stokes shift. Okay, that's important because most naturally occurring dyes have a low Stokes shift. It's just how nature is, right? But in flow cytometry, we're able to use more of the spectrum by having a lot of tandem dyes that we use. And those dyes are where we are going to take a chemical reaction and link two molecules that are fluorescent together, and that's going to force a fret. So it's going to expand the Stokes shift and allow you to excite something at a lower wavelength and get maybe 300 nanometers of separation or more between the excitation and the emission. These are not commonly used in other technologies that use fluorescent dyes, so you typically will only see tandems really in flow cytometry, much less so in something like microscopy. Okay, so looking at spectra again, and tandem dyes. What I'm showing you here is just a review of what a spectrum looks like and what the tandem molecule looks like. So we're going to get a donor excitation photon that is going to excite our PE. Because our PE is within angstroms of our size 7, we can take that emitted photon, push it to the size 7, and get a photon out that has much lower energy from the size 7 molecule. When you look at that in spectra, it looks like this. So these are the EX-EM wavelengths for PE at the top and PE size 7 at the bottom. The dotted line here is the excitation, and the solid filled histogram is the emission wavelengths. So nanometers on the bottom, and what we're doing here is we're looking at PE, and the filter that we use for PE, the optical band, is shown here in the blue box, and you can see that that's going to be a 58614. Whereas when we use a PE size 7 and we force that fret through through size 7, we have the same excitation profile and a little bit of emission from PE alone, but almost all of that excitation gets pushed through to the size, or that emission gets pushed through to the size 7 and is detected at 780/60 bandpass on a standard cytometer. You can see you do have a little excitation of size 7 by itself, and like I said, you do have a little bit of emission of PE by itself. So these two dyes will overlap with one another, and you will need to have some kind of spectral correction between them, but they're extremely easy to use together. Okay, so fluorochrome brightness. Brightness is the next thing you need to start to consider. It's basically the probability that a fluorescent molecule is going to emit a photon when it's excited. In a perfect world of just physics, this is determined by the extinction coefficient and the quantum yield. Unfortunately, we don't live in a perfect world, in case no one has noticed. And so brightness on the cytometer is really influenced by the efficiency of the excitation laser line to that particular fluor. So you often will be having a situation where your laser is not at the maximum, but it's at 75% efficiency on the excitation spectrum. And then also the sensitivity and range of the detectors. So brightness is a little bit harder to look up, but you really don't need exact numbers here. You just need to be able to group them into very bright fluors, intermediate fluors, and dim fluors. And this is going to give you a sense. And most panel building software and instrument or reagent suppliers are going to give you some information about brightness. So the way that we measure brightness on the cytometer is by looking at stain index. And stain index is basically a measure for how separate the negative peak and the positive peak of a stain sample are. So the stain index is the difference between the MFIs of the two peak divided by 2 times the standard deviation of the the negative peak. And what you're going to be able to do if you use this and you bought antibodies in every color, you could actually make a comparison. I'm sorry, if you bought anti-CD4, for instance, in every color, you could make a comparison of the dyes. And this is an example of, I don't know, 50 or so dyes, from those that are least bright to those that are most bright. You can do this on your instrument, and you should. That'll give you the ability to generate a spillover spread matrix, which you can actually use to characterize your specific instrument. And there's a paper here that can give you some more information on that. Okay, so your resolution is largely dependent on the amount of spillover, the amount of similarity between the floors that you choose, and how you're going to deal with that. So all of these are characterized mathematically by different kinds of matrices. These would include a compensation matrix, an unmixing matrix, a spectral similarity matrix. All of these are going to just give you a measurement of how similar a pair of dyes are to one another. It's important information, but for purposes of this talk, I'm not going to talk to you about how those matrices are different from one another. I'm just going to tell you that your panel design should think about what the compensation or unmix or spectral similarity or spillover spread matrices are going to look like with the panel that you use. So we're gonna walk through an example. Oh, this is a spillover spread matrix. This is an example of what you can do if you buy all those anti-CD4s. We can generate a matrix that's the inverse of the compensation matrix, and we'll tell you, just based on color coding, where I have dyes that are very similar, and this is where I expect that I'll have higher compensation or higher levels of unmix, and where I'll have spread that I can't correct for by those methods. Okay, so the SSM, like I said, is specific to your instrument and your floors. This kind of matrix gives you a sense of the directionality of the spill. So for instance, what I'm showing you here is that we have the primary dyes here and the secondary signal coming in here. And you can see that there's a lot of spill of, say, BUV 563 into PE. These two have very similar emission wavelengths. And that is directional. So PE does not really come into BUV 563, but the reverse is not the case. So when I design a panel, I'm going to want to know that because it's going to tell me how much that BUV 563 is going to impact the PE. And I'm going to know that the PE is not going to have very much impact on the BUV 563. And I should thank my friends at University of Chicago Flow Core. They have a great website. You should check it out. Okay, so when we look at co-expression, what we're looking at here is that when you have high spectral similarity between dyes, you want to have those pairs on dyes that are less related. It's going to improve your resolution, and it's going to mean that you have less sort of zone of uncertainty between positives and negatives. This is going to be very important for how well you can resolve things, and I'll have a couple of simplified diagrams to show you this in a minute. So examples are you can tolerate a lot of spill when you have markers that aren't co-expressed, like a CD19 and a CD8. When you do have co-expression, they should be further apart spectrally, so CD3 and CD4. With viability dyes, you're always going to only look at your viability negative or your live cells. So it's less important to have spill coming from your viability than from any other place. So I usually like to pick a viability dye at the low end of the instrument. I'm going to pick something and a UV 450 in order to exploit a channel that is maybe not that easily used and is going to spread throughout the panel toward the red. Okay, so the impact of this kind of spillover spreading, if I have high spillover spread and I have correctly compensated or unmixed data, what happens is that I have a marker A and a marker B, and if they're correctly compensated, that means that the MFI of the negative and positive populations for marker A will be the same. So the cells that express marker B and the cells that express marker A are going to have the same MFI, and they're going to have this sort of characteristic trumpet shape. Now that trumpet shape where the positives spread out a lot is where you run into trouble with panel design. So if you have high spillover spread and you have co-expressed markers, you end up with a situation like this, where you don't really know where your gate is. You can see you have a little bit of positive, but it's hard to really express that. If I plan my panel better, I'm going to have less spreading here in the marker A signal. And that means when I add marker A, I can see it separately. So that's my goal of panel design. Okay, so matrices. When I talk about compensation matrices, we're looking at the primary detector here, the secondary detector here, and as you read across, this is how much your first dye spills into the other, and as you read down, it's how much the other floors are going to impact the signal of the first one. So you can look at a bad panel, for instance, where I'm going to use 3 different floors off of the red laser, and those are always going to have more similarity. That's going to give me higher compensation coefficients, which isn't in and of itself a problem, but it also is going to give me much higher similarity between floors. And that's going to give me resolution problems like with the spillover spread that I showed you on the last slide. So on the other hand, if I took a good panel, like something like this that has BV421, FITSI, PE, APC, and I look at their compensation matrix, all of my compensation coefficients are quite low here. A similarity matrix is also going to show you that my dyes are very dissimilar. This is a good thing. I want to have the best resolution I can have. So if I'm looking at a 4-color panel, I want to have the least overlap that I can possibly have. Okay, so high spill is okay when our markers are not co-expressed and they have similar expression levels. And I hope this makes sense. These are non-co-expressed. My gates may overlap with one another, so they wouldn't fit quadrants perfectly, but I'm easily going to be able to separate those two populations. And it's not a problem that quadrants don't fit this data. It's just a fact of how multicolor flow works. Okay, so where is my high spill a problem? If I have co-expressed markers, that's going to cause my gate placement to be less clear, right? Do I want to have my gate here where I can kind of see the inflection point between the negatives and the positives? Do I want to have it here where it's all the way out of the FMO, right, in this pair? So in here, I'd be looking at maybe 2-3% positive in the FMO where I know I don't have reagent. And here I'm going to get a better measurement of the positives, but it's, or a higher number for the measurement of the positives, but there's always going to be uncertainty there. This is going to get much worse if you put the highly expressed marker on a bright floor and the low expressed marker on a dim floor. So we always want to avoid that. Okay, lots of web tools for this. As I said, I'm not going to go through these web tools for you with you. There are tons of great tutorials, but most of the reagent suppliers have their own either panel builders or spectral viewers, and these will link into their catalogs and help you find reagents. Some of them have some great background information on protocols and also what markers are relevant to your cell types. So I encourage everybody to explore the vendor websites and see what information they can get. There are also independent panel builders that pull in reagents from a lot of different suppliers. Some of these require a subscription, or at least they require a subscription for the functionality that you might want. And these include FloraFinder and EasyPanel. Both are great systems. All of these are great systems. They all have positives and negatives. I use what I use depending on my goals, but all of these tools to some extent. Okay, so simplest panel, got a 4-laser instrument, I want to do 4-color panel. I'm always going to choose the lowest emission wavelength detector for each laser. This is going to give me the fluorochrome that is most efficient with the lowest stoke shift off of each laser. So an example to this, and I'm going to have almost no spectral overlap between those. So an example of this, I have GFP positive cells. I want to add 2 antibodies and a live dead. So my antibodies are going to be PE and APC off the YG and red laser. My GFP is going to be off the 488, and I'm going to pick a viability dye somewhere in violet. And what you can see is this is my spectral overlap for each laser. I have no overlap going this way, and I have no overlap going this way. This is going to give me extremely high resolution data every time. Okay, so let's move on to an 8-color panel. Slightly more complicated, maybe twice as complicated, maybe a little more. Okay, so first thing is, what markers do I want to use? Well, I have a pretty small set of markers here, and they're going to allow me to talk about or to identify live T cells of both CD4 and CD8, as well as B cells and NK cells within my PBMC sample. So I'm going to classify those markers based on expression level. And there are some places online that you can find that expression level. There's lots of availability. You can also ask a friendly immunologist if you can find one A lot of immunologists know something about how bright their markers are. And you can look at the data on the technical data sheet, which will also give you a sense of how well separated the negatives and positives are in many cases. So I'm going to separate those into high expressors, intermediate expressors, low expressors. And then I'm going to take a pile of fluorochromes that I like, and I'm going to separate those into dim, intermediate, and bright. Okay. And what I'm going to do is I'm going to choose dyes here and here and here for high expression dim fluors, intermediate, and low expression bright fluors. And I'm going to put a panel together that is going to consider co-expression like we talked about and spectral overlap. Okay, so like I said, there are lots of good examples for answers for how you might get an 8-color panel. There's no right answer. There's lots of wrong answers. First, we want to look at what co-expression we have, and I've got this as both a table and a diagram, so you can get some kind of sense of this, but everything's going to be live dead negative. Everything's going to be CD45 positive. All my T cells are going to be CD3. Half of them will be CD4. Well, more than half. And less than half of them will be CD8, and then I'll have some NKs and some CD19 positive B cells. So this gives me a sense of what markers of what I have are going to be on multiple cell types, and that'll help me decide where I want to have spectral overlap occurring and where I want to avoid it. Okay, so next step, I've got my lists, I've got my idea of co-expression, I've got my bright fluors and my dim fluors, so I'm going to start to assign these. Now we're going back to the spreadsheet that I use here. Like I told you, I like a live dead blue somewhere in the UV range, so I'm going to assign that. Also, I always, when I'm looking at leukemia, or I'm sorry, immunology panels, basically, I'm always going to want to include CD45. It's a pan-leukocyte marker, and it helps clean up your data a lot. I like to use it in Alexa Fluor 700, which is one of the dimmest fluorochromes, because it's extremely highly expressed. It's at the far red, so it has less interaction with the other fluors than you might find if you put it into blue, right? So I've got the live dead all the way to the low wavelength side of the analysis, and that's because I'm going to look at only negatives, and I've got the CD45 all the way to the red laser on the far red, and that's because I'm only going to look at positives. Okay. Now I'm going to start to assign my other dyes, and this is going to be a matter of maybe seeing what I have in the fridge, but also spreading these out along the floor. So I've got 4 lasers here. I'm going to have 1 to 2 lasers, or 5 lasers here. I'm going to have 1 to 2 dyes on each laser. And when I pick these, I'm going to say, okay, I want to choose, there's not much spectral overlap between Pacific Blue and Brilliant Violet 605, but let's pick dyes that don't stay in the same cells. So I'll do CD16 and CD3. Similarly here, as we go across your circles, these are T cells, these are NK cells, and these are B cells. And I've split out how I'm going to assign those so that the spectral overlap that we do see between fluors that are included are really on dyes, or on non-co-expressed markers. Okay, so here's our 8-color sample panel as I set it up on our instrument, and you can see that I have 1 UV dye here. This is my viability. I've got my CD16 and my CD3 here off the blue. I've got my, or off the violet. I've got my CD4 and my 19 off the blue. Off the YG, I'm getting my PE here, and my PE size 7, and then I've got my Alexa Fluor 700, and you can see that's very dim based on the height of that peak. These boxes are our bandpass, and I want to make sure that the peak is within the bandpass, and in many cases, I don't need to stress too much about whether all of the peak is in the bandpass. Okay, so here's what this looks like from a similarity matrix standpoint, and as an 8-color panel, it comes to a complexity of 2.0. This tool is designed for spectral flow, but I find that it's useful for a lot of different kinds of flow. The principles for spectral flow and conventional flow are all largely the same. So this is really giving you a sense of how similar the dyes are, not necessarily something that's specific to a spectral cytometry experiment. So I've got a little bit of overlap here between my CD45 and my PerCP, or my PerCP Psi 5.5, which is my CD19 B cells. I don't think that's going to be a problem. So I'm going to go with this panel as is. My fluors don't have very much overlap, and my cell types are all separate on my little grid here. Okay, So now we're going to move to a bigger example. I thought I'd start out by saying that there are always compromises in a big panel. There's lots of bad choices. There's no perfect choice. For a big panel, you're going to start with the most rarely studied markers and the lowest expression markers. That's always my first set that I'm going to assign. The less rarely studied markers have fewer options that are commercially available. And the dim markers need to be reserved for the bright dyes. So those are the two groups I'm going to start out with. If I can, I'm going to save small molecule dyes like FITSI or Pacific Blue for intracellular staining, particular internuclear staining. It's stable and it's a good policy in general not to use a big light harvesting polymer on a internuclear stain if you don't have to. Okay, so after assigning those, you should be able to fill in your more common markers considering your expression levels, co-expression, and spectral similarity. Rarely used markers will have very few fluors. So just because you have CD4 PE in your fridge does not mean that you are going to be able to use it in your 20 to 40 color panel. It just won't work. So in a lot of cases, you're going to have to go ahead and buy new reagents if you want to increase the complexity quite a lot. You can do custom conjugates. However, if you do them through companies, they're very expensive. We do some in-house in my lab. There are some kits that you can do for them. There's a limited number of fluors available, but it's a good option if you're using an in-house antibody or some kind of antibody that you can't get conjugated. Okay, so now we're moving on to a spectral flow panel. This is going to be a 30, I think it's a 33 color panel. And when you look at this, the spectrum output looks much more complex, right? So this is our different lasers here, all the way from the laser incidence to the 800 nanometer or the end of the UV vis spectrum, or the end of the NIR spectrum. And you can see that I'm fitting in quite a lot of dyes here. It wouldn't be obvious to anybody's eye that these don't overlap too much to be able to use together. So we're going to use some mathematical tools to help us believe that before we spend thousands of dollars. This is only possible because there's been a huge revolution in the number of dyes that are available over the last even 5 years. And there's new ones that I find every day. So a panel I designed 3 years ago, I think I could make it better today than I could have been, right? All the time. So newer dye series are include the Brilliant dyes, Fire dyes from BioLegend, Sparks also from BioLegend, Star Brights from BioRad, E-Fluors and Nova Fluors from Thermo, Super Brights from Thermo. BD has their new series of real dyes. Cytec has C-Fluors. There's a million of them available. So think outside the box as you plan bigger panels. When you don't know these new fluors, there's a lot of information on the website. So they're going to give you the spectrum. They're going to give you a sense of how bright it is, right? Use that information as you start to plan, right? You should know that between companies, it's going to be very hard to compare brightness, but you can get a sense of it by cross-referencing your information. I think Fluorifinder does some of that, one of the panel design building things where it tells you some brightness information, but it doesn't interface with all suppliers. So there's, like I said, always pluses and minuses. Okay, so this is our more complicated example. We're going to look at a broad immunophenotyping panel for tumor infiltrating leukocytes. And in this case, I care about both lymphoid and myeloid cells. I want to know quite a lot about T cells. I want to know if they're effector memory, if they are central memory, if they're Tregs. I want to know if they express some of the checkpoint markers. I want to know if they're inside the tumor and activated. For B cells, I don't want to know too much about them. I'm just going to look for plasma cells and B cells. For NK cells, I want to know something about these. They use some of the same activation markers that you can use on T cells, so that's kind of helpful. I can maybe cut the colors back a little bit by exploiting that information. Myeloid cells, there are lots of different cell types. They're usually more complicated from a phenotypic sense, so you're going to need more markers to identify particular cell types for myeloid cells usually than you do for lymphocytic cells. So here I'm a particular interest is in DCs, in neutrophils, and in monocytes and MACs. And this is the list of markers that I have. And this, when I get my tumors chopped up, like Sarah and Leroy told you, and I get a nice cell suspension, this is going to allow me to really characterize that in a powerful way. Okay, so we've got our instrument configuration. This is the more complicated version of the same table that I told you about a minute ago. And this is our 5 laser 64 channel Cytec Aurora. And what I've done here is I've added some of my dyes in, and I have marked them for how bright the dyes that I have listed are. So the dim dyes are the ones that I'm going to look for my highly expressed lineage markers. And the bright dyes are what I'm going to save for either the or for low expression markers and the commonly used dyes like a FITSI or a PE. I'm going to save for my less studied markers, right? Because there won't be very much availability there. Okay, so I got all my markers here. I got them color coded. I've got B cells, I've got NK cells, I've got myeloid cells of various types. I've got T cells. I don't need to know everything about this at this point as far as what's co-expressed. I just need to know kind of in a broad brush what's co-expressed. Okay, so I'm going to take this, and as we move over, I'm going to pick out my low expression markers. First, I'm going to set my viability and my CD45. This is always a thing for me. I like this UV 450 for viability and my CD45 and Alexa 700. It just works good every time. And then I'm going to choose the ones that I have put in larger type because the larger type need brighter floors, right? So I'm going to choose all of these markers, and I'm going to consider what cells they might be co-expressed on for where I put them, right? So my Treg markers, for instance, CD25 and FOXP3, I'm going to put my FOXP3 onto FITSI. It's not a super bright floor, but it is a floor that is an organic floor, and I know that that conjugate works, so I'm going to go ahead and do that. And then my CD25, if I can find it, here I have on APC because there's just not very much of it on the cell surface, and I want to be able to define those cells, so I've got no spill between those markers, and I'm just going to put my CD4 somewhere where it doesn't interact very much with that. Same goes for the rest of these. These are less well studied, some of these, so I'm going to put these on here first, and you can see that the font colors are changing, right? I've got my T cell markers going separately from my myeloid markers, and I'm going to think about where I don't, if I have CD11C, it's not going to be co-expressed with CD68. So, because those are two different cell types. So it doesn't matter if they interact a little bit. It's usually iterative to go through this a few times before you get to a point where you're happy with it. But, every step, go back, look for problems. Okay, then I'm going to assign the remaining markers. These are the markers that are more highly expressed, so the ones that are in smaller font type here. So this is my HLA-DR and my CD11B, and these types of markers, CD44, 45RA, 62L, these are all going to be available in lots of colors. So this is where I'm going to fill in with my more obscure dyes that you may not have heard about, like my Spark dyes. We're having good luck with those lately. These real dyes from BD seem to be pretty good. So I'm starting to work those into a lot of panels. So when I get this set up, what I'm going to be looking at is I'm going to look across and say, is there any co-expression here? And I'm going to look down and say, is there much co-expression here? I want to know, okay, these are close, right? BUV486 and BV480, but they're on two different cell types. This is a CD8, this is a CD138. I've considered that there may be overlap and spectral or spreading, spillover spreading between those two, but they're not going to be on the same cell type. So I end up here, and you're just going to assume that for the magic of PowerPoint. This is what this is going to look like as far as emission profiles. So again, we have the UV channels, the violet channels, the blue laser channels, the YG. This is from the Cytec cloud, which is at this point I will have moved into the Cytec cloud to work on this panel. And you can see what my all of my emission wavelengths look like. And if I click on any of those, I can see how broad it is, whether it has any secondary peaks like this noise and stuff here, where I'm going to expect to see some spread in my panel that results from that. Okay, so now I'm going to evaluate my panel as I designed it. This is the same sheet I showed you right here. I've done this before, so my complexity index here at 33 colors is 16.5. This is a nice place to be. You want to generally not really exceed the number of parameters in the complexity, right? So if I have a 30 color panel and I go much above 50, I'm really going to have serious resolution problems, even if, it will work, but it might not get you the information that you need. I'm going to pull up the similarity matrix. This I've shown you before. This is, I don't know exactly how the math works, but it's a measurement of how similar any two dyes are. This is useful, especially when you don't have many, many years of doing this in your head. And it will tell you where you have higher shading and a higher number. It's going to tell you where you have spill. So For instance, here, I can tell this is 87% similarity here between BV421 and E4450. So I'm going to come over and I'm going to look at this and I'm going to say, oh, well, good, because I put CD69 on BV421. That's a low expression marker and it's only on T cells. And I've got CD16 on my E4450. So I should be okay with it. I'm going to consider that fine. So still working entirely in the Cytec cloud here, not an endorsement, just works well for me for Cytec panels. Okay, I'm gonna look at other cases in my spillover spread matrix. Now this here is off that center line, right? So these are the dyes that I know are gonna be kind of down the column here as we go. And so if I see a lot of high numbers that aren't on the center line, I especially wanna look at those. And here's one in particular where I have 81% similarity between APC and BUV661. So I'm going to look at that and I'm going to say, okay, are these, I didn't expect that to be quite as much similarity as it was at 81% between a BUV that emits at 660 and APC that excites off red and emits at 660. What are the markers there? Well, I have PDL1 on APC and I have CD45RA, and those are both T cell markers and they could be co-expressed. So this might be a problem. Here's what the spectra look like for this. This is the BUV661, and you can see that its peak is a UV here. But everything out past the YG looks extremely similar to APC, right? You can see that those are not going to be easily distinguished. And this is going to make them more similar than most other pairs. So what am I going to do to fix this? Okay, I'm going to look at, I've got this whole row in the column, right, where I've got CD45RA, 64, which is a FC receptor, CCR7, which is on T cells, PDL1 that's on T cells. And when I look at similarity into that APC and I worry about what's going to impact my resolution on PDL1, I can pull up similarity scores between each of these. And you can see that the similarity to APC is really only very high for BUV661. If I look at BV650, same wavelength off the violet laser, much lower, right? PE640, same, much lower. APC obviously is 1 because it does 100% similar to APC. So I'm going to look at this and I'm going to say, okay, I don't want to have two T cell markers here on BUV661 and PDL1. Instead, I want to maybe swap these two and have a myeloid marker that's only on myeloid cells in that noisy BUV661 channel. So I'm going to swap those two. It's not going to change anything as far as my similarity matrix, but it is going to give me a stain index reduction matrix that is going to be significantly better based on what I know is going to be positive and negative. And you can see here that your stain index reduction matrix, which this one you can get at a couple different places, but this one is also out of the Cytec cloud, is going to show me where I have these spill events between these 4 dyes that I just showed you. And I want to be super careful that where I see a stain index reduction, they're not co-expressed, and hopefully they're on very different cell types and will be in a different place in my data analysis as I get that far. Okay. And I was just going to say again, you note that my viability here in UV450, I like this channel because it does spread into everything, but I never look at dead cells, which are the positives. So as I look at the matrices, they're all going to kind of give me the same information, right? So the next talk, I think, is Peter Mage, and that will be very much interested in what the numbers mean here. For me, I'm really looking at color codes for the purpose of this talk. Okay, so at this point, I'm going to make that switch, like I told you, from C45RA and the 64. I'm going to look at every other pair with high spread and verify to myself that this is going to work before I spend my hard-earned grant money. And then I'm going to make any other necessary changes that I can imagine that may reduce sensitivity. Now I'm ready to order. Great news. Okay, so once you got your reagents and you've got your cells, you're going to start to technically evaluate your panel, and that's going to be testing it with pilot experiments, including FMOs. You can often use tissues that are not super important to you, so you could use a PBMC sample from human or a spleen sample from a mouse that's less something that you've got months invested in than your experimental samples. I'm going to identify problems that I have with resolution and possibly problems that I have with spectral unmix or compensation, and I'm going to troubleshoot those problems there. Resolution problems should only be done after I know that I have good unmix and compensation. So get that right. Repeat your single stains if necessary in order to get your spectral unmix okay or your compensation okay, and then look at where you're having resolution problems. And you may have some iterative solutions here. You can switch brighter dyes for dye or switch out for a brighter dye that's in a similar profile. So I've done this successfully with like a Percy PSI 5.5. If I go to a Brilliant Blue 700, I get much better separation. I swap the antigens if I have to for a high spill situation. And at the worst case, I'll cut unnecessary markers that are causing me problems. So at this point, once I've done all that, then I can start to look at my mice, my tumor-bearing mice, and my important experiments that are really the part that I'm going to publish. This is just the getting it working. Okay, and when I can do that, I get good resolution on my UMAPs, I get nice spanning trees, my data is beautiful, and my scientific questions can be answered. And I think that's all I got. Yay.
Okay. Okay.
Our next speaker is Peter Mage, and he is going to talk to us about compensation and spectral unmix. He's coming to us from BD, and he has been in the field for quite a long time.
6 years.
6 years, and
Feels long.
always has great ideas. So please enjoy.
Thank you very much. Okay, cool. I think that this is working. Let me get my little laser pointer here. Thank you so much, Karen, for the kind introduction and the excellent overview of panel design. I think that was a really accessible but comprehensive way of looking at it and a really important step as we'll talk about today. My talk is about building your spectral intuition. What I mean there is when, say you get invited to give a talk on best practices for unmixing, compensation, and spectral flow, and you don't actually know what those are. You can instead think about what you do know, which is kind of understanding how spectral cytometry works. So really what this talk will be about is not so much what the best practices are. I'll give you some resources to refer to and go back to as you're planning your experiments, but instead understanding why those best practices are the best practices and having a conceptual toolkit in your head that you can use to help answer some of those questions and to help infer and figure out what's going on in the lab when you run into problems, because you will run into problems. So, I like being here and coming to these because I am an engineer and a physicist by training. Many of you here are biologists, so we're kind of mixing the chocolate and peanut butter, hopefully making a Reese's cup and not just a mess. But I think that sharing our perspectives can be really valuable. My path to get into cytometry is a little unusual, actually. So in grad school, I really focused on making biosensors that could measure molecules in the blood. I did a postdoc that got me on in front of an ARIA, sorting some weird synthetic particles based on shape. And that made me interested in flow. And so I joined BD about 6 years ago and broadly work on computational design techniques that we can apply to instruments, to algorithms, to reagents, to panels in order to build really big experiments. And so kind of thinking about how do we get to ultra high parameter flow. We recently submitted a paper on a 50 color panel that was mentioned earlier today. So how do you get to that level? Well, by making tons and tons of mistakes and doing a really bad job for a long time until you figure out how to do it. So I am always intimidated giving these kind of presentations because most of you know a lot more about what I'll be talking about than I do. But when I first got into flow, a really helpful text and kind of source for me was Howard Shapiro's book, Practical Flow Cytometry, which many of you, I'm sure, have seen on your core manager shelf or have maybe even looked at yourself. But at the very start of the book, Howard presents his laws of cytometry. And the 0th law that comes before even the first one is that there is no magic. In other words, everything happening in your experiment, in your instrument, in your software, there is a physical reason for why that is happening. You may never learn what that reason is, but you can trust at least that there is a reason that everything is happening and that everything is the way it is. So today, what I want to do is help us kind of learn to dispel magic when we think about spectral flow, and we'll do that by thinking like an engineer, right? Breaking down the problem, applying mental models, approaching it from multiple angles, and getting frustrated, but eventually not giving up. Okay, so some of these questions that might come up about spectral flow, there are always why questions, like why should you listen to me? I can't answer that. Why should you care about spectral flow? Why are these best practices the way they are? If you're a Talking Heads fan, you may ask yourself, well, how did I get here? If you're me, you have to ask, how should I design a cytometer? But I think a lot of us are really here because we want to know how to do good spectral flow cytometry, right? You want good data. You want those beautiful results that Karen was talking about. You want your PI to get off your back. But before you can answer these questions, I think it's really helpful to think about the what, right? To develop a deep understanding of what we are actually doing, what we are talking about when we think about spectral flow. So our talk today is kind of going to break out like this. We'll talk about the why it's important to think about spectral flow, and we're really going to spend most of the time thinking about the what. So building some mental models for understanding how spectral cytometry works, and then we'll apply those mental models to think about how we should best do it. And we're going to spend a lot of time talking about 3 different mental models for spectral flow that we'll get into, thinking about it as a process of signal transformation where we reveal invisible molecular features, thinking about it as a mode of artistic expression, which I'll explain, and then finally thinking about it as a mode of mathematical mapping, the details of which will really resonate with everyone in their post-lunch kind of blood sugar crash. Okay, so part 1 is the why. Why should we care about this? I think it's helpful to frame what we're talking about in terms of why we care about cytometry at all. Fundamentally, what a cytometer does, right, is measure one cell at a time. If you measure them quickly and if you do it carefully, you can measure and sort millions of those cells all in one experiment. And by doing that, you can learn multiple things about each cell and compare those things to other cells. And you can start to learn, what are those cells doing? How are they related to each other? What is their fate? But as all of us know from spending time in the lab, you don't just learn by measuring more stuff, you learn by measuring it correctly and by measuring it well. So of course, how much I can learn per cell depends on how much I measure per cell, right? The number of features. And so if I measure more features versus fewer features, I can resolve kind of more detail. But it's not just the number of things I measure, it's the quality with which I measure those, right? So if I measure with higher resolution, I can tell finer detail and infer kind of more subtle biological patterns. So in flow cytometry, both quantity and quality are going to matter. The challenge is that quantity and quality are very directly and inextricably linked to each other in flow. And hopefully you got some of that from Karen's examples as we built from simpler to more complex panels. You now have to start thinking about all of these compromises and trade-offs that come in. And the reason we think about that a lot now is that the field has really made the shift from conventional to spectral flow, like we talked about a little bit this morning. Historically, you could add colors to a panel or add colors to the field by just filling in empty parts of spectral space. You go scuba diving off the Monterey coast and find a new fluorescent protein and some algae, and that's how you get PE and APC. Or you develop a new type of dye technology that takes advantage of a violet laser, and that's how you get the brilliant violet dyes. And that process led to kind of high parameter conventional flow, right? Those of you who have used, say, a symphony from BD, there's kind of this 25, 28-ish color level where we were at for a long time. But in the last 5-10 years, there's really been this shift. So instruments are no longer detector limited. You can throw as many of those bad boys in there as you can afford. And dye technologies, like we just talked about, have really expanded the palette of colors that are available. But if you think about it, it's not an easy answer where to put those colors. There's no easy spectral space left. I'm not going to belabor this point because I think Karen did a good job explaining it. But as we think about going to higher and higher panel complexities, we now inevitably have to start dealing with the fact that my dyes overlap and they're going to be co-expressed. And we're going to get this loss of resolution occurring as that happens. So where before the question to go to higher parameter flow was adding more dyes and more detectors, now the question is, how do you add the right dyes? How do you add the right detectors in the right part of the spectrum to maximize the quality of your data? Okay, so we know why we're talking about this, or at least I know why, maybe it wasn't clear. Let's now talk about what are some models we can use to understand flow cytometry. And like I said, we're going to break it into 3 mental models that I have found helpful for understanding this as we sort of have worked through this at BD. Okay, so let's think again about the basics, right? The way a cytometer measures a cell is by, well, first we're motivated to learn about proteins on the cell. Those proteins are frustratingly invisible, so I can't just look at them. So instead, I label each protein or some other type of molecular marker using a unique fluorochrome or color that's attached to an antibody. And then I'm going to measure that colored light somehow. And the amount of colored light I measure is going to reveal something about the amount of each protein that was there to varying degrees of quantitativeness. So to understand what spectral flow is doing, we need to really think about this process where we go from a labeled cell to some information. Well, if I break this out step by step, I'm kind of going through this process of signal transformations. So first, I've encoded my molecular information with these colored molecules. I'm then going to excite those molecules and measure the resulting photons that come. Of course, the total signal I get is just the sum of each of those individual fluorochromes that's on my cell. The cytometer is then going to sample that with some number of detectors and convert it to a digital signal. And then I'm going to do some kind of math to try to infer from the total signal that I measured, this purple curve, what the individual contributing amount of each of those dyes was. And that's what's going to tie me back to something that's essentially meaningful in biological space, right? Number of cells with a certain number of molecules in a certain pattern. Spectral flow, compared to, say, conventional flow, really influences how we go through 2 steps in this process, how we convert colored light to a digital signal, and how we convert a digital signal back into some biologically meaningful data. So if we think about spectral and conventional flow, which I think most folks might be more familiar with, like Karen said and some of the other talks have mentioned, we're really doing the same thing. Conceptually, they're not so different. We're labeling those cells with fluorochromes, measuring the total signal, trying to infer how much signal came from each fluorochrome, and identifying cells with that information. There's really only 2 essential differences between spectral and conventional flow. In conventional flow, in the hardware domain, we use 1 detector per fluorochrome that I'm planning to look at, which is why most of us are familiar with the idea of a GFP channel, a FITC channel, a PE channel, et cetera. In spectral, it acts more like a spectrometer, which you may have used in, say, a chemistry lab. We're going to have more detectors than fluorochromes and roughly just sample across the entire spectral space, again, usually in the UV vis to NIR region. And finally, on the software side, we're going to use different math to go from my whatever set of digital intensity measurements to something that's more in kind of molecular units that I care about. So in conventional flow, we call that compensation. In spectral and mixing, we call it spectral and mixing. And the details there are not interesting, and we're going to talk about them later, so you can look forward to that. Okay, but the important thing is not the steps that go in. The important thing is that every time we're transforming signal from one domain to another, the quality of that transformation is going to impact the quality of my final unmixed data, my final biological data. And if you're familiar with machine learning or with AI, maybe you're familiar with the concept of feature engineering. So it's this concept that we're going from some latent layer that contains our molecular information, and we want to get that into whatever the cool algorithm we're using is today. This is a little cartoon of a neural network, but ultimately we want to get some meaningful phenotypic insight out about our sample, right? Well, every step in this chain you can think of as a layer that we are transforming our signal with, and it's going to impact our final data. So we're encoding our molecules as fluorescences. We're then translating that to some number of detectors that are converted to an electronic signal and a digital signal, and then we do some sort of unmixing to get our final biological signal back that feeds into our actual analysis. And every step in this chain involves design decisions that either you have to make when you're designing a panel, or that someone at a vendor has to make when they're designing an instrument that impacts the final quality of that data. And so just like there's this concept of feature engineering in machine learning, where you're sort of creating features from your raw data that will be useful for something like a classifier, say, we can think of every step in this measurement chain as also feature engineering. Panel design is a really clear example like we just talked about. So again, I won't go into too much detail here, but it's an important step where we're deciding how to map invisible biological information to a machine interpretable signal. So that seems like it should be pretty important. And indeed it is. If I look at two different panels, the same underlying biology actually measured on the same instrument from the same sample that was just split in two, I can see in my top panel that I'm able to get pretty good resolution for these different populations on some T cells. And if I use that different panel, a different choice of encoding or feature in this transformation pathway, I see that actually because of spillover spreading, because of insufficient separation, I've lost a bunch of information. In other words, this second choice of a panel was not engineering a maximally informative feature set for my experiment. So our goal in all of this is to engineer maximally informative data to answer the biological question that you have. And we're going to have to think about that carefully at every step in the signal transformation pathway. Okay, so you may be thinking, that was a lot. It's still pretty soon after lunch, and that wasn't fair. We will take an art break now to think about this from another angle that maybe will help some of these concepts make some more intuitive make some more intuitive sense. Excuse me. Okay, so some of you may be into painting or into finger painting. You may have kids who are into finger painting. I think there's sort of a fundamental joy that's revealed when we think about the fact that we can create new colors by mixing other colors. That's just fun, no matter how old you are, no matter who you are. So if I take an example of, say, mixing blue and yellow paint, I know that I can get green. But I also know that by mixing those paint colors in different amounts, I can get different new colors out. So if I take the same amount of blue, but a little bit less yellow, I get something more like a turquoise or a cyan. In fact, this is a task that can make you TikTok famous. If you want to get into this, act fast, because this may not be available as a career path for a while. But there's a whole category of TikTokers who look at like a paint chip and can, with crazy accuracy, tell you exactly the individual paints you need to take in to match that tone exactly. There's a whole like ASMR thing to it that I try to not get too involved in. But this is a whole genre. If you're interested, you can look these up. But this reveals something about how our brains are wired, that we can look at a new color, and if we know what other colors were available, we can kind of figure out what colors went into it. In other words, if we know what colors are available and we see a mixed color, we can mentally unmix what colors went into it. So if I show you green and I know that, say, your kid only had tubes of blue and yellow finger paint and the outside of your Tesla is now a lovely green, you can probably figure out the relative amounts that they used. And again, if I show you cyan, you can infer something about the relative level of each color that went into that new color. Well, hopefully the analogy is sort of starting to become clear here. Just like we can create new colors by mixing primary colors in different amounts, we can think of cytometry as taking individual paints, except instead of paint, it is a single fluorochrome like PHITC or PE for my blue and yellow colors here. Each of those has their own individual color that's going to mix to give some new characteristic spectrum based on their relative abundances. And then I could just eyeball this if I know what the individual spectra are and try to infer, okay, how much PHITC and how much PE went into the total new color that I measured on that cell. Except I'm not going to do that. I'm going to use a cytometer to do it. We can even extend this a little bit and start thinking about, okay, intrinsically, it's kind of harder to mentally unmix two colors if they were really similar to start with. So we know that APC and Alexa 647, this was kind of a classic example to show what spectral cytometry could do initially. If I show you a mixture of those, it's hard to tell how much of each of those dyes would have gone into the mix. So if I imagine two red colors like Merlot and Fire Red, and I show you another color in the middle, it would be hard for us to infer whether it was more Merlot and less Fire Red, or maybe less Merlot and more Fire Red. If we did a poll of the room, we would probably get something like spread in our results that illustrated. That's kind of getting to be a tortured analogy. So let's think if we can get to a more rigorous model here. Well, indeed, right? The way that we interpret color as humans has some analogies to the way that a cytometer works, right? Our eyes have individual types of photoreceptor cells that are sensitive in different parts of the spectrum. Their responsivity actually kind of smears into each other, but we can approximate it with this cartoon here. And those receptor cells work with our brain to interpret a given input spectrum as a single new color. Well, in a cytometer, like we just said, we use multiple detectors to sample that spectrum. But instead of representing it as a new color, we represent it as a series of numbers, which is not as interesting, but computers love that stuff. And then the cytometer and computer work together to take that spectrum and report the new color as a series of other numbers that are meaningful. Okay, so let's tie it back to what we've been talking about with cytometry here, right? If I know the primary colors that were used, I can infer their relative contributions using this kind of reasoning we've been talking about. And those relative contributions, again, are what I ultimately care about for my experiment. Compensation and spectral unmixing, they're just the names for the specific type of math that we use to do this process, okay? So I promised we would talk about that math. I can see that some of us are still awake, so that's really great. You're going to get the most out of this for sure. But I think it is actually helpful to talk about the math for how compensation and spectral unmixing works. First, because you're going to have to use it when you do compensation or spectral unmixing, so it's better to sort of know your enemy than be taken by surprise. But also, there are some insights we can get from how the math works that will help us do some pretty powerful things later on. So when we talk about compensation and spectral unmixing, all of this revolves around a type of math thing called a linear mixture model. And I'll explain what that means in a second. But basically, it's a way of formally describing how these signals from different fluorophores combine on my cell to make a final total signal. So I could think of looking at each of my dyes in isolation. Let's say I have a blue, a green, and an orange. I'm going to go from take my first dye, look at how much of its signal falls into each of the detectors on my toy cytometer here. And then I'm going to convert that to some kind of digitized signal that's in discrete amounts, because ultimately that's what a computer is going to work with. So let's say I convert that spectrum into these little balls. I have 5 for my first detector, 1 for my second detector, and 0 for my third detector. If I want to know how much signal I would see from 2 of these blue molecules, all I'm going to do is take that signal per dye, multiply it by the number of blue molecules that I have, and that's going to give me the total contribution from that dye. I can go through and do the same process for each of my other colors. I won't walk through all of it for the sake of time, but you can trust me that these numbers are all real for this toy example. And then the total signal that I measure is just going to be the sum of the total amount of each individual dye, right? Of course, again, the cytometer doesn't know what color a photon was necessarily when it comes into a single detector. So it just knows that it saw some total number of photons in each of these detection bands. And we can represent that like this. So now let's think about how we might describe this problem using math, which is always more fun. I'm going to use this little animation and kind of rotate the problem. And I'll admit, I stole this from Dave Novo, who showed it at the first cytometry conference I ever went to, and I really liked it. So thank you to Dave. Basically, what I can do is rotate my problem and keep all the numbers and values the same and put this all into one line. So I'm going to say my blue spectrum times the number of blue molecules plus green spectrum times number of green molecules plus orange times number of orange molecules gives me my total. Because this is math, it has to be less fun. So we're going to start condensing this and sort of putting it into a more compact notation. There's still sort of color and joy here. So we're going to go all the way to just making it numbers. This bottom version is a lot better, clearly. But we can see that all I've done is kind of use a more compact notation here where I make what's called a matrix, multiply it by what's called a vector, and get a new vector out. And here I'm just showing that in the more condensed matrix notation that linear algebra uses. And again, it's worthwhile at least knowing some of those terms because that's where some of the language of matrix inversion and things like that come from when we talk about compensation and unmixing. I can give these specific names because there's nothing that math likes doing better than taking numbers and replacing them with letters to make it even more confusing. And so these are the terminology that are used in some other cytometry education materials online, Rui Gardner and some others. So I'm just using those for consistency. I'm going to call this matrix R for reference matrix because it contains my reference spectra for each dye. A is my abundance vector, so how much of each fluorophore I have. And M is my mixed or measured signal vector, tells me the signal I saw in each detector. Okay, so all I've talked about is essentially how my signal showed up. And you could have gotten that by just measuring your sample. So we didn't really care about that. But in reality, we care about something else, right? The things that I know in a cytometry experiment are the individual fluorophore spectra, which I get by going through a single stain workflow. And I know the signal that I measured from my fully stained sample because I just measured it from my fully stained sample. But what I want to know is the underlying amount of each individual fluorochrome, because again, that's what ties back to the amount of each molecule on the cell that I care about. So I can describe this expression for the math as what's called the forward problem, right? It's describing where the signal came from. But what I can do using, again, linear algebra is solve what's called the inverse problem, which is where I want to know what the sources of my signal were. Can I calculate that using the other knowns in my equation? Indeed, I can, and this is how that works out. So you calculate something called the inverse matrix of your starting spectral matrix, and that's what we call conventionally the compensation matrix. I just calculate that, multiply it by my measured detector values, and get my compensated data out. So that is literally what your cytometer is doing when you are performing conventional compensation. In the sort of more olden days, when going to two-color flow was a big deal, they actually used analog circuits to do this compensation in real time. I think Bartek actually explained that to me quite a while ago, which I thought was really interesting, and I hadn't realized that. This is how this is sort of done today using digital systems, which most of us are using at this point. Okay, so let's talk about spectral. It's the same thing. It really is almost identical, except in spectral, like I said, the essential definition is that we have more detectors than dyes. So you can see I now have a fictional cytometer with 4 detectors, and I've represented what the signal for each of my dyes would be there. I can represent the forward problem in the same way. So kind of the shape of this problem looks the same, except this matrix is a rectangle instead of a square. And I can, again, frame the inverse problem the exact same way. So when I'm going to solve this problem, when I'm going to do spectral unmixing, I'm just going to be looking at this lower right-hand set of equations, except instead of using a compensation matrix, I use a different matrix that doesn't matter, but it's called the pseudo-inverse matrix of that spectral matrix. I just said matrix more times than I intended to. I apologize. Okay, so that is all an extremely compelling and interesting way of thinking about flow cytometry. But there's another way. This is the value of going through the math, is that when you look at the mathematical representation, it unlocks a whole bunch of other ways of looking at the same problem, some of which have useful solutions or concepts that you can apply to your problem. So one example of that is that these linear mixture models, they aren't just representative of like solving these kind of sets of equations and making matrices. They actually have a really clear geometric meaning, which is it lets my data go from one space to another space. And that may not sound interesting. Sorry, but I can explain what that looks like in an actual data example. So here I'm showing the compensation case. What compensation does, literally mathematically, this is exactly what it is doing, is it maps my data from one two-dimensional space, if I have a 2-detector system, where my data is correlated because signal from one fluorophore gets measured in the detector for another fluorophore, and it stretches that space out to make the data no longer correlated. So that's why we use compensated data, right? If these were on 2 different markers that I wanted to tell apart, I don't want to see false correlation that would suggest there was biological co-expression there, when in reality, it was just from my measurement. So compensation is mapping between 2 different 2D spaces and kind of stretching my space when I do it. Spectral unmixing is actually doing the same thing, except we go from a higher dimensional space, however many detectors I have, to a lower dimensional space, however many colors I have, right? That's fundamentally what spectral unmixing does. Now, I tried really hard in PowerPoint to illustrate a 78-dimensional space. Didn't work. So we kind of have to use a 3D and a 2D space to get some human intuition here. But again, I'm taking the same data here, but I've brought in data from a 3rd detector, and I've plotted that for these same 2 populations. And you can see that, indeed, I have signal in a sort of 3-dimensional space here, but all of that data actually falls on this 2-dimensional plane within the data that when I apply spectral unmixing, stretches that higher dimensional space to a decorrelated low dimensional space that tells me something about my biology. It's okay if that doesn't seem relevant, because there are tools that we can use from this concept of linear mapping that will help us in a little bit. Okay, so just to illustrate this a little bit more clearly here, I can think of spectral cytometry as, again, this process of transformations that are also mappings between different spaces. So let's say that the molecules I care about are in some initial lower dimensional space. Say I have 2 markers I want to look at. I'm going to convert that into a fluorescent signal that exists in a higher dimensional space, shown here as 3. And then I'm going to unmix back into a lower dimensional space that will hopefully reflect the underlying molecular space that I care about. And the important thing is that the map that dictates how that kind of rotation and stretching and compression process goes, that is the spectral matrix. That set of single stain spectra, that is what describes how signal maps from molecular space to detector space. And that pseudo inverse matrix that's calculated from the same thing describes how data maps back from the high dimensional space to the low dimensional space. So whenever we're doing cytometry, we're just mapping between these spaces. And it turns out that you can map and do a better job or a worse job, which is what we're going to talk about. Okay, so let me summarize all of this, right? We can think of the way a cytometry experiment generates signals as this forward problem. I go from cells with labels, I measure those, and they are mixed together. I can represent that geometrically as mapping from a low dimensional space to a high dimensional space, and there's math for describing that. The inverse problem is where we just go backwards, and this is what we do when we do spectral unmixing. So I have some mixed total spectral signal. I'm going to unmix that and get my lower dimensional space. I can represent that geometrically here. And again, there is math that describes it, so we can do math to this to learn new things. Okay, you made it through all 3 models. Now we just have one section left, which is how the hell do I use any of this to do something useful? And there is an answer, I think. So let's dive into that. So again, let's tie all of this back together. I've introduced spectral with the concept that we're transforming our signal, and the quality of our transformation at every step in that pathway impacts our final data quality. And I've shown how we can also represent this as a process of linear mapping, where we apply a linear mixture model. And it turns out that the language of that math can give us quantitative tools to understand the quality of our unmixed data. And the reason it's important to think about quality and unmixed data is because, well, A, that's the actual data you're going to look at, and that dictates how much you can answer your biological question. But B, it's kind of justified by everything I've been talking about. All we're doing here is applying a model that may or may not accurately describe our system. So anytime you apply a model, you want to make sure that it is actually accurate and that you are making the correct assumptions and that it is giving you results you can trust. So again, this linear mixture model, it's a way of describing pretty much correctly how a cytometer generates signals. In order to be useful, we're kind of leaving out some details by making certain assumptions. How can we quantify that those are correct, that we're actually doing the right thing? Those of you who have done some statistics or machine learning hopefully have kind of heard of some of these concepts where we want to think about quantifying the quality of a model in terms of bias and variance, or we can also call these accuracy and precision, right? This is just sort of ripped off from how Wikipedia illustrates this, and most of you are probably familiar with it, but I'll quickly illustrate it here, that we can think about having low bias and low variance, where we're very correct, and on average, our predictions are all really close to each other. If I have high bias, I always am sort of predicting consistently for different inputs, but they're on average pretty wrong. I can be right on average, but have a really wide spread, or I can be doing a pretty bad job on all fronts and have a wide spread in my answers and also on average be wrong. That's how I feel a lot of the time just in life, but not in cytometry. Okay, so bias again tells us something about how wrong we are on average. Variance tells me how spread out our answers are for individual, let's say, events or cells. We can apply the same concepts to troubleshooting problems in spectral unmixing or compensation. And the reason I'm presenting it this way is that I think this actually gives us a systematic way to debug unmixing and compensation problems that tend to feel pretty mysterious. Essentially, any problem in your panel's unmixing can be described either as bias, where you have something like over or under compensation, or what people are now calling unmixing errors, which I'm illustrating down here. Or it can be described as having high variance, where you have too much spread in your data, right? Sometimes you have both happening at the same time, and that makes it kind of hard to debug what's going on. But all of the problems you might see fall under one of these 2 categories. That can help us then think about troubleshooting our unmixing or compensation results in a more systematic way. Okay, so let's describe what this means a little bit more in concrete terms for those of you who are using spectral cytometers. Again, bias tells me something about how accurate our mapping is on average. This would manifest as sort of population level errors in our unmixing. So this is traditionally what we would call over or under compensation. And when we're looking on sort of a bi-X plot like this, it tends to look something like this, right? The most common culprit here is that something went wrong in your single stain workflow, or something has happened so that the spectrum of dyes in your sample no longer matches the spectrum of your single stain controls. And we'll go through that in a moment. It is possible and in fact good to avoid bias, right? You should be attempting to have an experiment with no measurable bias in your unmixing. And you can do that through careful controls and through a careful workflow in most cases. This is generally a symptom of an experiment level problem where you need to think about how all of your data is being processed. Okay, variance on the other hand is again talking about spread. How much spread is there in my data? This is something you can think of as event or single cell level unmixing error due to random noise, because we have noise coming from all different kinds of sources. This is going to mean we can never perfectly fit every single cell in our sample. And the most common culprit that's going to matter when you're doing sort of multicolor or spectral flow is spillover spreading, right? Where you have signal from one dye causing this increase in variance of another dye's unmixed signal. Unlike bias, you cannot avoid variance entirely. It is always going to be present. In fact, there's a well-known trade-off between bias and variance when you're thinking of a model. We don't have to go into those details, but your mindset should not be to have no variance in your data, because that's impossible, but it should be to minimize it through panel design, through setting up your instrument correctly, etc. Variance is an unavoidable property of cytometry data itself, but something we can try to work around. Okay, so quickly, I'm going to go through some ways to think about how to fix your problems when you see either bias or variance in your data. Again, this whole pathway of signal transformation I've talked about, it's assuming this model, this linear mixture model. And that makes two key assumptions that are actually in the name. The first is that your signals behave linearly, which is actually, I have a hard time explaining that, so I'll do that in a slide. Don't worry about it if that's not clear. And then it second assumes that your map is accurate. That means that if the signal really was generated with some matrix R, the matrix you're using for unmixing had better actually match those real spectra. So our assumptions are going to be wrong, and we'll have bias in our results when one of two things has happened. Our data isn't actually linear, or our map, our spectral matrix, is incorrect. What does this actually mean for our data to be linear? It really means two simple things. First, the signal that you measure is proportional to the amount of fluorophore. That is usually how this is going to work. One notable case where that doesn't happen is if your signal goes off scale on a detector. That means now that the signal you're measuring has saturated on height and is no longer providing a proportional response on area. So this is why it's important for your data to all be safely on scale within the linear range of your detectors when you're measuring either your controls or your fully stained sample. The second thing that linearity means in a technical sense that's a little bit more subtle, but explains some things that we see in practice, is that the expression or presence of one fluor does not affect how another fluorophore behaves. So if I have PE and FTSE together, the signal I measure from the two of them together should just be their individual signals added up. There shouldn't be any other thing going on. These two things are the strict definition of linearity in a mathematical sense, but there are real cases where they can be violated. So I just mentioned that signal saturation, having events off scale, violates this first proportionality requirement. Any interaction between dyes that are expressed together that changes their intensity or their spectrum as a result of being together, that is a nonlinearity. So if two dyes are threading into each other and changing their emission spectrum, if one dye is quenching another dye, those are all things that will actually lead to unmixing errors potentially, because the system is no longer behaving linearly. Now, having a wrong spectral matrix is a little bit more self-explanatory. Really, there's two ways that it can be wrong. You can have fluorophores that are present in your sample that aren't present in your matrix, so unaccounted for signal, or your reference spectra may not match the actual spectra on your sample. Again, what causes that to happen? Well, the most common, like 90% of cases, is that there's an issue with your single stain analysis. So maybe your gating was not exactly correct. Your positives and negatives came from different scatter populations and have different autofluorescence. Maybe your single stain was dimmer than your fully stained sample, so you're missing some dim parts of the spectrum in that reference spectra. There are things that can happen where you did your whole workflow correctly, but it turns out that the fluorophore actually changed its spectrum in your fully stained sample, maybe because there was a different buffer present, or because you were using beads for your controls that have a slightly different microenvironment on their surface than cells. And then the most common case where you've actually just missed something entirely is if you're handling autofluorescence. Most cytometers give you the option to either include autofluorescence as an unmixed color when you're doing your experiment. In a lot of cases, there's not very much autofluorescence on your cells, so it doesn't matter. But if you have an autofluorescent sample where you're looking at dyes that are similar to the autofluorescence spectrum, not accounting for that in the unmixing will also lead potentially to this bias presence that you'll see. Okay, and I actually want to reiterate the paper by Laura Ferrer, who's formerly in New Zealand. She's actually working at BD now, which is awesome for us and I think for everybody else, that really goes over the best practices in a very practical sense of how to run these controls, how to design your experiment to avoid these types of problems in a spectral experiment. So could I have fulfilled what my talk title is by just putting up this slide and telling you to read Laura's paper? Absolutely, but it's okay. We're 45 minutes in already. Okay, for the sake of time, I'm just going to very briefly go through this variance explanation, just because I think that it is relevant to when you're thinking about going to the really high parameter range in these spectral panels. So we're all familiar and have seen slides like this where we can lose our biological resolution because of spread and noise coming in from different sources. Everything from the instrument detectors to free fluorophore in my solution to laser background. The thing I want to emphasize here is that when we do spectral unmixing, there is actually a situation where the unmixing process itself can introduce or actually amplify the amount of noise that is present in my unmixed signals. How does that work? Well, I showed this concept of mapping before. Turns out if you go through and do a bunch of math, it turns out that you're actually introducing noise inevitably when we go into this detector space, right? And that noise sticks around when I go back to my unmixed space. So even though I can do this mapping, I cannot get rid of noise that gets introduced, which is inevitable. There's math to describe how that process works. I'm not going to talk about it, except to mention that the complexity score that Karen mentioned in her talk and that you'll see on all of these sort of spectral panel design websites, it's a mathematical thing. It's called the condition number spectral matrix. You can look that up on Wikipedia if you're interested. It just describes how badly that matrix is going to amplify noise that came in from your measurement in your final unmixed result. So a higher number means you're getting sort of more amplification of noise in your unmixed data. Okay, why that matters is when we think about this feature engineering process, when we think about making really high complexity, high plexity panels, we're going to inevitably have a high degree of spectral overlap. And that can lead to something called unmixing dependent spread. And this is going to be essentially the limiting factor for how big a panel can get currently. What this means, I can illustrate with a quick example. So our collaborators were trying a couple years ago to do a 50 color panel. And when they went from a 20 color staining with their backbone panel to the full 51 colors, they saw this kind of catastrophic loss of resolution. And you might say that that was due to spillover spreading from all those extra fluorophores, which is totally reasonable. But in reality, they could take the same raw data from their 20 color stain and unmix it with the 51 color matrix and see again this huge loss of resolution purely from the mathematics of that spectral unmixing process. So this leads us to define this unmixing dependent spread concept. It's a change in variance of our unmixed signal when the same raw data is unmixed using different spectral matrices that either contain a different set of fluorochromes or different numbers of fluorochromes. So to illustrate this, I could show you 3 different examples of fluorochrome pairs. And if you're going to pick one of these to go into your panel, you would probably choose the one on the left. Maybe the one in the middle is acceptable. The one on the right, absolutely not. These are all actually just from PE and BB 515. Same raw data, just unmixed with different unmixing matrices with different complexity scores. And I can see as I go to bigger and bigger panels where I start to get way more spectral overlap, all of the dyes in that panel, I get this deterioration in my data quality that purely comes from the unmixing process that it eventually makes my data unusable. There's a bunch of math behind this that I'm not going to talk about today. There will be a couple of talks at Cyto about this if you'll be there, and we're working on a paper right now so that you'll be able to go back and look at this if you're interested. What I can say is that figuring out some of those details was a part of helping us develop this 50 color panel, which I'm just showing a super high level overview of here. To really hit home and kind of summarize that understanding this math at a pretty deep level can help us unlock some new technologies that I think can move the field forward, right? So using some panel design techniques that were drawn from that math, we were able to come up with a 50 color panel that actually has a lower complexity score, here shown as 40, than some of the previously published 40 color OMIPs. Some of that is because the dyes available now, even compared to a couple years ago, are much better and make this easier. So it's not that those panels aren't good. It's just that by paying attention to this noise mapping, we can actually potentially push the field even further, which I think is pretty neat. So with that, let's just tie it all back together in 30 seconds. All of this matters because how we transform these signals in the spectral pathway impact the final quality of our data. We care about having high quality data because we learn more by measuring better, not just by measuring more things. So ultimately, I hope that this has given you at least one little thing or a visual that you can take back to your spectral or compensation experiment and maybe apply when something is going wrong, and can help ultimately solve some problems. I'd like to thank some folks at BD and our collaborators at the Hutch in Seattle who helped with developing some of these techniques. And I think we'll do questions at the roundtable at the end. So thank you very much for your attention. Am I introducing Lisa? Perfect. Thank you. Appreciate it.
Really nice way to explain a lot of those concepts.
I appreciate that. Yeah.
All right, everyone. Can you hear me? Yeah? All right. This is Dr. Lisa Nichols. Oh, did you share your screen? Dr. Lisa Nichols is a really wonderful person. If you haven't met her yet, please take the time to meet her. She received her PhD at some point from the University of Virginia. I don't know when, but yeah. She signed up a letter and got it. And she has worked in industry setting as well as academic settings, and she now runs the Stanford Shared Facts Facility as the director. I was lucky enough to work under her. She is a fabulous mentor, a fabulous educator, and really useful information, so make sure you listen.
I paid her to say that while she was there. So thank you very much for that lovely introduction, Meredith. And thank you to Texas for hosting me. I know this is the FlowTex meeting, but old stomping grounds, hard to give up old habits, bad habits. I'm not sure which. That's to be evaluated, right? So there has been a lot of really great talks today. There's been a ton of information. Anybody here do any assays besides flow cytometry in your experiments? Nobody? Come on. This is the last talk of the day, and I cannot be the only one talking. So this is going to be very, very interactive. I know you don't like that. The people in the back are going to be especially subject to questions, just FYI. So this is a lot of time to spend on one assay. And as we've seen today, there's really a lot of kind of very detailed information that's gotten in there. So I'm actually not going to really tell you anything new in this particular talk. What I am going to do is hopefully put it all together in a little bit of a workflow and really kind of see how all these pieces fit together and what are kind of some of the very kind of nuts and bolts things we need to do to get our experiments done. So let's go ahead and start off. We're going to talk about data evaluations, controls, and cleanup. We are hopefully going to be moving forward in the PowerPoint presentation. There we go. All right, so a recap of today's sessions really have talked about a number of different things, starting with the scientific question. In our very first, Dave and Joel introduced flow cytometry, and they said that the first one came out. Anybody remember what year? Oh, that's good. So I think the expectation at this point is that we should be able to just take these scientific questions and move right from, this stuff's been around since longer than I've been alive. We should be at the point where I can just hit a button, the little AI thing will pop up and give me that, right? This is where we want to be. And I think that it's very exciting to hear a talk from Peter, who kind of helps you see the possibilities of, hey, if we have enough understanding of how these instruments work, how these fluorochromes work together, and the math that's behind all of this and the science that's behind all of this, that even my undergraduate who's working in the lab with me maybe can get to this point, right? That's the pipe dream. Okay? What I'm going to talk about is before we can just jump in, push a button, and get this sort of data out of there, though, this is where we want to be. Even for very simple experiments like a single color, do we have GFP? Do we not have GFP? We're going to have to do one more step, and that's really to take a very hard and critical look at the data that we collect on these instruments. And so we're going to talk a little bit today, or a lot of it, about the controls that we're going to need to make all this happen, as well as some of the data cleanup we need so that we can get those nice clusters out that actually give us that meaningful data. So I think he talked about bias, and so basically the accuracy and the precision of your data. And what we'd like to do is get to a point where we can get to that and feel confident that we got to that point. So I believe it was Sarah that mentioned earlier today, garbage in, garbage out. So it wouldn't be a talk without pulling in an internet meme. Basically, what if we get all of this information, we do all this sample prep, we design this assay, we spend a whole lot of time and a whole lot of money buying 30 fluorochromes to put together our assay. How do we actually know that we don't have garbage in and garbage out, and that we actually do have reasonable data and reasonable conclusions? So we're going to do what I like to call pre-data analysis. I know last year at FlowTex, you guys did an entire huge workshop on high parameter, high dimensional assays, right? We're not going to do that today. This is actually going to be, what do you do with that data that you collected before you get there, and how do you get to that point? So there's two pieces of it. One of them comes way before the experiment. None of this is new. You've heard about all of this in pieces throughout the talk today. The controls that you need when you design it, what controls are necessary to interpret it. And then the second piece, am I using that high quality data at the end? So a good friend of mine, colleague, Jen Wilshire, used to call it forensic flow cytometry. How do we know that we have cell viability, singlets, good data, et cetera? All right, let's just jump right in. Like I said, this is going to be practical. This is going to be short. It is like 3 or something, 3 o'clock. All right, coffee's kicking in. All right, we're going to go in this. I want you guys to stop at any point, ask any questions. The first one that everybody always asks me, I do a ton of teaching, right? You guys are super busy. You have at least 4 other assays going on in the lab. You want to get your data. You want to ask a very specific question. And you want to get out. Are all of these controls necessary? Experimental controls, assay-specific controls, single color controls, and gating controls. We're going to start with the first 2. And again, this is way before you even touch the flow cytometer. These were discussed at the very beginning of the talk when they talked about all the different applications. Well-designed experiments will have a negative control. What are the baseline conditions for your experimental conditions? A positive control. How do you know the assay worked, right? This is the first time you've done flow cytometry. You got a whole bunch of dots on the screen. How do I know that that's the best that signal can be and actually tells me something that's biologically relevant? An experimental sample. I think this is the part that the scientists have the easiest time with, right? This is the question that you actually want to ask. Your scientific question, what is the outcome of my experiment, right? You all came here to ask a question. It was not, how does a flow cytometer work? It was, what happens in cancer cells? Why are the immune system not doing its job? What happens if I give this drug and I treat this person with X? Why is it that old people get shingles and young people don't? What happens with these iPSCs? Are they really what they're supposed to be? Because they don't look like much in the dish when I look at them in the microscope. These are all the questions that you guys want to actually ask. In this part, we don't really need to spend any time on. This is kind of what you naturally do. Okay? But you can't just go and look at data like this and answer a question. So somebody tell me, is this a healthy or a disease? I don't know. You have to have something to compare it to, right? You must have that control. It's critical for you to have some sort of baseline. Otherwise, these are clusters and they're really pretty meaningless, even if you could drill down and see what types of cells those were in them. Okay? This happens to be actually healthy all the way around. This just happens to be peripheral versus central lymphoid tissue. So. Experimental controls. I took a cue from the first group, CFSC. We want to ask a question. Look, are the T cells able to actually see antigen? This is a very common question. Does this happen in the tumor? Are the T cells just going and are hanging out? Are they not seeing it? What's going on with these guys? Do they actually respond? So the question that I wanted to ask in this particular assay is, hey, are they actually able to see it? This is your scientific question. The assay that you're going to select maybe is, hey, T cells, I expect them to divide when they see what they want. So this is my assay that I selected. We talked about this earlier today. He did a great job explaining, as Jake was telling you about CFSE, right? Throw a whole bunch of green on them, and as the cells divide, they get less and less and less green. Okay? Again, experimental control. This is easy. You guys are going to compare what the T cells do when the antigen is there versus the knockout. Pretty easy question, right? We do this assay. We throw these super green cells into the mouse, and then we look, okay, well, in the knockout, there's nothing for them to respond to, so this looks good. They're still nice and bright green. That's in this direction. In the wild type, I don't see anything. So Jake said they were going to do this. They're supposed to divide. They're supposed to not be green. Well, they're definitely not green. But they have this other marker which should identify them. I don't see them all. Anybody else see them? But I have an experimental control, an assay control that basically says, well, maybe I missed mouse when I stuck the cells in it, right? Maybe I just missed. Well, I have this internal control. This is an assay control. You guys need to build these into your experiments. Okay. For some reason, flow cytometry, I guess because it came out in 1954, thank you. We kind of think we don't need to design and think about this stuff, but we really do, right? So we need to think about the fact that, yeah, I know I injected because I mixed 50-50 cells that have the marker versus cells that don't have this other marker, right? So, well, here's the cells that have the marker, but the green ones up here, those are gone. So we got to step back and say, well, okay, this is not the expectation. I expected they would divide, and this is what Jake said they would do. I don't see them. So we need a better control. We need an assay positive control, something that says, hey, I know that the T cells are going to see this particular antigen, and they're just going to go nuts, and they should do exactly what Jake promised. We're throwing you under the bus all day long. It's okay. So what would it look like if these things actually proliferate? So one thing is we're going to go a couple days earlier, because you notice that really bright green cells, they were kind of sitting only mid-scale. And so I'm going to look at day 3 instead of day 7, because they seem to be losing their dye. So I'm going to take that variable out of there. And the next thing I'm going to do is I'm going to throw in a viral infection where I know the T cells are going to respond exactly as I expect them to do, because that's what they do in the context of a virus. So this is our positive control. Sure enough, we see a whole lot of division, and these things are just piling up gangbusters. Now we can ask our scientific question. And as you can see at this time point, same conditions as we had the other day, just an earlier time point. You can see that, in fact, these guys are dividing, but they're not building up gangbusters. They seem to be dividing and then just kind of going. Okay. So this is how we go through, and with a nice set of assay-specific and experimental controls, make sure that we're actually asking and answering questions in a way that's meaningful and can actually interpret this. So it's just something to think about as you're designing your assays. This is just one type of control. And again, I think this is the one that's most natural to scientists, even if you've never seen a flow cytometer. Another example of this, we talked a lot about membrane potential, mitochondrial potential as an indication of cells going into early apoptosis. So here we go. We're going to use TMRE. This is going to light up our mitochondria. They're going to be able to retain this dye at very high levels. They're nice and bright red-orange here. And then if the cells are apoptosing, going into early apoptosis, this is going to go through the leaky membranes, and the level of that dye should work. So again, my scientific question, I got this knockout gene. I expect maybe these cells are going to start to die, that they're not going to survive quite as well. So I'm going to look at the wild type mitochondria, and here we go, TMRE, and our wild type, that looks completely normal. But I really kind of expected that I would see, my hypothesis was, for this experiment, that I would see more apoptosis here, but this is 38% versus 24%. Is that the best signal to noise? Maybe that's just as good as it gets, and that's how this assay works. And I think, again, he also indicated this. I need an assay-specific positive control. If I really do get a really nice apoptotic response, and indeed do lose this mitochondrial potential, we're going to treat with a drug that actually forces that to happen. And here you can see that we have a significant shift in the signal. So again, kind of taking those pieces that I think we know as a scientist, but I cannot tell you how many times I have a consultation, and these kind of real basic things seem to be skipped over pretty frequently. So they're really critical. All right, they provide a basis of comparison. They provide information on whether or not an assay has worked. All right? And again, you could apply this to any sort of assay you're setting up, not specific necessarily to flow cytometry, but something to not forget either. All right, the second piece we're going to talk about are controls for spectral overlap. So if you had to name the number one thing that every biologist hates, okay, there's two. I think Peter hit on it. Math, you guys love math, right? And this is the Achilles heel of all flow cytometry, is all of this spectral overlap. I'm not going to spend any time on this, but I would point out to anybody, I get these questions, well, I put them all on different lasers, they're not going to have any spectral overlap. And I will remind everybody that of all these fluorochromes, remember, this is in wavelength on the bottom. We're going from about 350 out to about 850. That's the usable space that we're using right now. There are very few of the fluorochromes that are less than 100 nanometers wide in their emission. So that means between 350 and 850, we have how many? That's 500 nanometers. On the guys in the back, we're expecting you guys to pop up here, right? So that means that maybe 5 are going to not overlap appreciably, okay? So just expect that there's going to be overlap until proven otherwise. That's just a good rule of thumb to start with. And then, if you find out you don't need the control later, I don't think it's going to cost you anything then, right? If you needed that control, however, because even very low parameter experiments are going to have a bunch of overlap. If you need that control, you're going to be in a lot of trouble, because trying to interpret how many CD4 cells and how many CD8 cells in this that has a lot of overlap and I didn't have the right controls, versus this where we can actually correct and understand and calculate those overlaps so that we can figure out how much of each is present, is going to be critical to you being able to use that data at all. You cannot really get any information out of this data. All right, so let's, oh, that's a weird animation. My tubes all disappeared. So these cells are just magically standing. They used to have tubes. I don't know where they went, but we won't worry about that. So we're going to talk really quickly, and I'm going to make this pretty quick because, again, this has been mentioned, and I think that if you've done any flow cytometry, you're at least vaguely aware of this. You have to have those single color controls. And Peter showed you with the mathematical model as well as all the other models that if you have a cyan, right, a cyan bucket of paint, and you need to know how many of those blues and how many of those yellows are in it, you'd better know what blue looks like and you'd better know what yellow looks like, right? So that's going to be the single color, the GFP, the RFP. And you also had better know what the background looks like, so autofluorescence that's coming out of that background, okay? So we can't just load the GFP all by itself. It's a small molecule. It's not going to trigger the instrument, right? So what we have to do is attach it to something. I don't care whether you attach it to cells or you could attach it to beads, but in either case, you have to have something that carries that through and it has its own background signal for autofluorescence. That background, it seems obvious, should match whatever's on the positive. Yes? You should do it every time because it's how the instrument measures it at the time that you use the instrument. So under your current experimental conditions. The things that can change in an experiment day to day, the cytometer can change. Most modern cytometers do have QC programs that do some adjustment for the minor variances on a day-to-day behavior basis. But unless you are really well in tune with what those are and how well they're functioning, you shouldn't assume they do. The second is the dyes in the cells that you use also can change over time. So if you have a tube of something that's a tandem dye and the PE is handing off to the size 7, when they're really nice and tight, you excite that PE, you get mostly size 7. They start to get a little bit old, or your undergraduate left it on the bench. They didn't tell you because they just shoved it in the fridge. They really don't want you to know. Now they're sitting like this, and you're going to get a little bit more PE and a lot less size 7. So if you don't bring those controls, again, you end up in a situation where you just can't use the data. So you either need to have a way and know enough about how the instrument's functioning to be able to do kind of a recovery afterward and bring in the appropriate control afterward. And for that to be accurate, you really need to know a lot more about your instrumentation. And I don't want to speak to that because how your core monitors the day-to-day behavior of the machine versus how some other core does that or your own particular lab does that in order to make sure that on Monday and Friday, the machine is giving you the same result. There's going to be error in there. So short answer, bring them every time. Okay. All right. It does need to be the exact dye. And again, old PE size 7 versus new PE size 7, a different lot. Those could give you different experiments or different results. And I think as Peter mentioned, they should have a similar intensity as the fluorochrome as used on the fully stained sample. So we'll talk more about this in just a minute. Okay. So we've already talked about whether we really need them. The answer is yes. Now the question is, well, I don't know. Do I really need cell controls or do I need bead controls? Which is best? Which is going to give me the best answer? Lisa, just tell me what the answer is. I get this a lot, right? Just tell me what the answer is. Well, realistically, it depends on a bunch of variables, which Peter again went into, right? So this is, again, not new information. So if the beads and the fluorochromes on the coating of the beads give you the same fluorescence as that same fluorochrome on the cells, and that can be calculated appropriately, then that works, but often cells do a better job of that. So I know that if I put a fluorochrome on the cell environment and I do a single color, that when it's in the multicolor environment, it should behave similarly. Now, he brought up a caveat situation where there's maybe fret between molecules, but that's probably going to be the best control we can get. So our assumption is that the cells should behave pretty much the same for their fluorescence if they're single color or multicolor. Yes? That one? That situation? Yes. Yes, the exception. So cells are usually the best control for doing these calculations, except for the point that Virginia just brought up. And that is when your cells are really dim or when your cells are really rare, right? Anybody working on stem cells here? Great. What's the frequency in the bone marrow? What do you do? Oh, what's the frequency there? Low. So the amount of cord blood you get, I believe, is under 15 ml, right? Yeah. So you would have to run a real large amount of a huge percentage of your sample in order to get enough events here to do a good calculation. So counting statistics come into this. I can't take the average of 5 cells and subtract a background from them, and then get you a good estimate for what that dye is. And the whole point of these single color controls is that you can estimate what the dye looks like in the primary detector, as well as all of the other detectors that those photons are potentially getting into. So in a situation where you have to run 500,000 or 5 million cells in order to get any sort of measurable signal, then cell controls are a bad idea. Similarly, if they're so dim, they don't really separate out from the background very much. Maybe I'm forced to draw my gate right here, and I have overlapping distributions of my positive and the negative. Again, it's very difficult for the instrument when it does the math. Your positive control now has a mixture of positive and negative in it, or maybe the number of amount of signal off of those is statistically, you're going to get very, very low, poor statistics off of that if that dye amount is very dim. So in these two instances, this is a great control, right? We have very bright signal, and we can calculate those dyes and subtract this background pretty easily. So that is really the exception. So I keep talking cells versus beads. Does anybody not understand what beads are? I'm going to go through this super quick. The idea is basically that manufacturers have put out a whole bunch of beads that are essentially vehicles for antibodies that grab your antibodies. So if you want to put 10 different colored antibodies on your experiment, you're going to put these beads into a tube. They're going to capture your antibodies, and now that bead background is now going to also be positive for whatever antibody fluorochrome you put in there. So I can have PE positive beads, I can have FITC positive beads, simply by just coating these beads with the antibody where they get captured. They typically have either in a separate tube or the same tube, the background control. So beads that won't capture those antibodies and therefore would be the negative control for that. There are 2 that I know of, GFP and M-Cherry for fluorescent proteins that are also coated onto beads so that you can use those as controls as well, rather than having to make single fluorochrome, single fluorescent protein controls. But if you are working with something like M-Kate, DAPI, anything like that, then you really can't use bead controls, or you're going to be stuck with cell controls. So sometimes beads actually aren't available. The advantage of beads, these are awesome. You put them in a tube, the dropper, throw some antibody on top of them. You get a really nice bright signal. The disadvantage is they can be too bright. So in our facility, we have the best signal to noise set up on all of our detectors. The intention of that is if I have a very dim signal and I have the best signal to noise on that detector, if my positive is just above my dim right here, I should be able to resolve that. And none of our users have to worry about increasing the voltage on that detector any further to get better resolution, because it just won't help them. Now, if with that very sensitive detection, your beads are sitting way out here, the other assumption that Peter said had to be correct was that you have to be using the linear range of the cytometer. So if your beads are smoking hot and bright, and this guy is off scale, you might have to decrease the sensitivity of your cytometer just to get those beads back onto the usable range of the cytometer. Okay? So you can't use something that is in the nonlinear region. And as I mentioned before, the overlaps that are calculated on beads, they don't always provide accurate compensation on cells. So whether it's because the fluorochromes behave differently on the plastic surface versus on a cell surface, or whether it's because the fluorochromes are, there's autofluorescence background that in the counting statistics isn't being calculated. It's disrupting the calculation for that dye across all the detectors. In any case, if you apply a bead matrix, so if you use all bead controls, maybe I have a 5-color experiment, and I generate a matrix, compensation matrix, or a spectral unmixing matrix on those beads, and I apply it to cells, sometimes it just doesn't work very well. So let me show you an example of this. This is just per CP on the y-axis versus some other fluorochrome, and you can see that it definitely spills from this detector here, which is the primary detector, into the secondary. And when I make my bead-generated compensation matrix, you can see that if I apply that matrix back onto the beads that generated it, it looks really good. There's definitely some spillover spreading, but the means of the negative channel, so it's negative for APC, these are centered, and then this is the spillover spreading here. If I take that same bead-generated matrix and I apply it to my cells, however, you can definitely see that this is overcompensated. There is no such thing as negative fluorescence. So it is okay for an event to have a negative fluorescence after compensation. So in other words, this could be 0 plus or minus some error. And here it looks like, I don't know, plus or minus 20, something like that, or 200. So there can be a negative fluorescent event, but there shouldn't be a mean that is negative. So this is overcompensation. If instead I take a cell-generated matrix and I do the same exact reciprocal experiment, you can see here that the cells are now properly compensated. They're centered over the mean, so these are orthogonal to one another, whereas the beads are actually slightly undercompensated. In other words, the mean is over here and not centered over the negatives. Okay? So this is how you're going to evaluate whether that bead matrix is actually working correctly on your cells. All right. So the most common compensation problems that we have, besides the using a bead matrix completely on cells without testing it, are these are the things that people most likely come into my lab and say, I can't unmix my stuff, because they forget that those viability dyes, they get so hung up on the antibodies. I'm doing a 10-color experiment. That means 10 vials of antibodies. And they forget that the viability dye is a color, right? Because it's not an antibody, so it's like in a different part of your brain, and you did it in a separate step anyways, right? So they forget that, and then they add that to all of their samples. So now your PE-only control now has PE and DAPI. So that's the mistake number one. The second is they use the wrong background for the single color. So remember I told you for the beads, you now have a compensation bead with an antibody tagged on it. So I have PE plus a bead background. And then I have these amine reactive beads. They're still beads, right? Now these beads are intended to grab the viability dyes. They're not even the same size, folks, okay? So if you take the compensation beads as the background for the amine reactive beads, you're making an assumption here again, and that assumption is wrong, right? So those do not have the same autofluorescence signatures, so you're subtracting the wrong background. It would be similar to having HeLa cells and lymphocytes in the same experiment and saying, hey, my HeLa cells are GFP positive, but I don't have any GFP negative HeLas, so I'm just going to subtract the lymphocytes. You know, you wouldn't subtract 1/5 from 1/4 without doing something with the denominators, right? So that's kind of the same analogy for subtracting the wrong background here. All right, and again, this has been mentioned before, you must treat the controls the same, especially with these tandems. If you fix the single cell controls, you should also fix. Sorry, YouTube. Everybody hear me now? Okay, cool. So anyways, you don't want to treat them separately. So if you're going to fix the experiment, right? So you're going to do some intracellular staining. If you need to fix and perm, you're going to want to use that same fixative and the same sort of buffer solutions on the single cell controls so that things like the tandems and the fluorochromes are all fluorescent and behaving exactly the same. And then my favorite is if you're going to use fresh antibodies on the cells, You may not want to use compensation controls that have been stored for a week. This is some data actually Meredith generated when she was in the lab. So beads are not always bad, and I don't mean to give them a bad name. Sometimes they work really well. So this is an experiment where I have these two colors here. It doesn't really matter what they are. I mixed them with cells. You can see the compensation looks good. These are orthogonal, right? So the means are centered around the negative here. And I mixed them with the ultracomp beads. Still looks beautiful. So compensation beads do work. You just need to check to make sure that they did work. They look beautiful when they're mixed with the unmixed with the one comp beads. I don't know what beads these were. They were sitting around in the fridge though. Doesn't look so good anymore, okay? So bead controls can and do work. I don't mean to give them a bad rap, but that doesn't mean that just because they worked on Friday means that they're also going to work on April in 2-3 weeks. All right. So the whole point of these single color controls is that it's going to enable accurate calculation of each of the fluorochromes in the experiment. That is their entire purpose here. For now, we're actually going to also add a couple of other controls in here. One of them is the unstained. So we already need that unstained control in order to know the background to subtract from the positives. I'm going to show you a little bit later how I also use that actually to assess resolution. And then the final control that we're going to want here for gating are going to be the fluorescence -1. This has been mentioned already a couple of times, so I'm going to go over this just really quickly. Is anybody here using FMOs routinely? Awesome. So you guys are a very experienced group, so this is great. We'll go through this just super quick. So gating controls, I get asked, do I need the FMO controls for compensation? Not really. They're actually used to determine where the gate is. So even in this very simple, this is just a simple GFP sort that we were setting up for somebody. And I'm going to have you guys analyze this data for me. So they bring in the cells and they tell me that they put something in them. They're supposed to be GFP positive, and they want to sort the GFP positive sample. So where do you guys think I should draw the gate? You work in a core. Anybody think this looks good? I think it looks pretty good, honestly, right? I mean, I'm not trying to be weird here. Like, this looks like we got some positives and some negatives relative to one another, right? So maybe only 50% transfection efficiency. I don't really know. That's my assumption. That's my assumption. All right, sample number 2. These are all just different variations. I don't really know what they did to the cells. Maybe different genes that they stuck in there. Again, I don't know. If you ask me without any other information, I'm going to be like, yeah, that looks positive and that looks negative. Yeah. We're going to put the gate there. Or, oh, I don't know. That's the same gate that I used in the first one. So maybe I should scooch it over a little bit because now maybe the negatives go there. The positives are there. Sure. Everybody okay with that? How about this one? So now I'm starting to really think these are all supposed to be kind of the same. So I don't really know what to do at this point. I'm like, I mean, is these really the negatives and the positives? Are they all positive? Here's the thing. This is your science experiment. You shouldn't be guessing, right? We should be making controls. So the proper control, I can already see you guys. Well, if you just line them up, we can be able to see this a little bit better, right? And indeed, these all look kind of positive, but they're not quite as positive as this positive, and that's my positive control. So I don't know if these are negative. We'll see. So I did bring in a control. Does this change your mind about any of the gates? Changes my name. This is the untransfected control. And the only question I still have, and again, the key here is you need to talk to the people doing the experiments. You guys need to think about this. This is a great negative control with one assumption. What is the assumption I'm making for this negative control? Bingo. Did anybody hear? So she said transfection didn't change it. So in other words, did this unstained control have no transfection and therefore was less autofluorescent? And therefore, this is the real autofluorescence of a GFP negative. So what do I need to ask? I need to ask, did this one get transfected? Did it get transfected with an empty vector that doesn't have GFP? Now, I know almost 0 people that actually do that. I would say maybe 5% do that, right? And that's okay. And it's a lot of work. So I'm not trying to, say that you absolutely have to have that. I'm saying you're making an assumption here. And then you have to now ask your scientific question. It may very well be that you really don't care about these cells. You have plenty right here. Just sort the bright ones and let's move on. Or it could be that it's actually a problem for your assay to have so many cells with so many copies of the GFP that you really don't want the bright ones, and actually maybe the negative ones are better. So it really comes down to a conversation and an understanding of what the scientific question you want, what the purpose is of your experiment, and therefore what the best control is, and whether it's necessary to take the effort to make an actual transfected control. Okay, so just realize the limitations of the assays. What was Howard Shapiro's quote? There's no magic. Okay, so let's just remember good controls because they really make all the difference. Gating controls take out the guesswork. All right, last place I'm going to talk about gating controls, and this again came up, spillover spreading and why gating controls can be so important. If I just use the unstained, untransfected, whatever as my control, I would say that right here is where I would probably put the mark for whether something was CD3 positive or not. But when I look at my single stain controls, you can see these negatives actually shift up a little bit. So this looks like a very clear positive, very well separated from here. And in the context of a fully stained sample, where do I now draw this gate? Now, I would say I don't draw this gate. What do I really need to do instead? This goes back to Karen's 40 color panel. Is it 30 color? I don't remember now. This right here, your single cell controls will give you a lot of information about the resolution. So if you do your staining on beads, you're just going to have beads that are positive and negative for a fluorochrome, and they will help you with compensation, but they won't help you with answering this question here, which really gets to the resolution of your experiments. So by yourself, you should be able to maybe get whatever percentage this is, but when you're in the context of the fully stained, you're no longer going to be able to clearly gate the same percentage that you had in your single stain control. So having single stain cell controls, another major advantage of them is that you can actually use them to determine the resolution of your experiment as well, or to at least give you more information about it. And the final piece that's also been mentioned, fluorescence -1. In a case where there is spillover spreading in a well-designed panel, you still can use the fluorescence -1 to figure out positive versus negative. So this is a panel where we're looking at CD45R positive cells. There's quite a bit of spread here from some antigen that's now spreading, is co-expressed on these cells, but it's not co-expressed on these cells. So when we go to look at, I think this is PDL1 on this axis right here, you can see that if I just use these cells here that had all the co-expression and I drew this straight across, I would be actually cutting quite a few of the positive PDL1 positive cells out of my calculation for this population over here that did not have so much spillover spreading. So fluorescence -1 controls. So again, this is where all of the colors in the experiment are there. -1 can really give you a lot more information. So for this population, we can see the positives coming out here. And we're probably losing a few positives down into this right here. But over on this side, you can see that we really would have lost most of them if we didn't have that FMO control to draw the gate. So FMO controls are not really compensation controls or unmixing controls, they're gating controls. As are your unstained can also can help with that, but won't give you all the answers. So 4 types of controls. All 4 are really necessary. Your experimental variables answer your scientific question and whether the assay performed as expected. You have spectral unmixing controls that tell you how much of each individual dye is there in a multicolor experiment. And then you have gating controls, fluorescence -1, as well as unstained, that help you to determine and see where you should actually be saying, hey, this is positive and this is actually truly negative in the context of my experiment. So that's the gating controls. I think the last piece of this is going to be super quick. And it's kind of an idea of, look, you guys gave me all these things to think about today. I've got 30 colors, I have spillover. How do I actually know that all of this worked? So we're going to do just very quick. This is about 4 slides, some forensic cytometry, and we're going to tie this all the way back into your cell prep, as well as your panel design, as well as your compensation. So maybe 6 slides. So the first one, Sarah and Leroy talked a lot about sample prep. It is the key to everything. Again, garbage in, garbage out. If you look at this here, we're looking at a scatter plot. We draw this around kind of our scatter, our cell gate here. We're going to look at singlets here. Now on this diagonal, if we look at area versus height is where we should find singlets. And then off to the side, we're going to find a whole bunch of doublets. So why does singlet gates matter so much? Because this is a single cell assay. So unless you're specifically asking a question about how two cells behave when they're stuck together, you're going to be wanting to look at one cell at a time so you can get the phenotype of that cell. So if we then do a live dead gate on this in order to analyze it, we can see that if we look at the single cells down here, I'm just going to skip this part over here for now. If we look at the single cells down here, they have a different distribution than these doublet cells that are right here. So the non-singlets went down to here. This is the live and the dead down to here. Over here, we have the singlets and the non-singlets, and you can see that we get a lot of garbage in here. This is actually drawn wrong. I do apologize. This is totally drawn wrong. All right, these guys here are actually single cells, and these are the doublets, and you can see that they have a very, very different distribution. These arrows should be coming off of the singlets going to here and the doublets going to here. Over on this side right here, we have actually gated for live versus ungated. So over here, we have a live gate, and over here, we have ungated. And you can see that by getting rid of the dead cells that are positive for the zombie near IR, we get rid of all of these things like aggregates, doublets, et cetera, throughout the assay. So you do want to clean those up. This is another assay where I had somebody come in and they said they brought 10 million cells and they wanted to sort these double positives. So this is their sort gate. Okay, sample prep is going to be key here. Brings me 10 million cells, and out of this 95% double positive, she got only 100,000 cells out of that. That is a lot of time and money wasted. So what happened to her cells? So we kind of went back and just looked at her flow cytometry plots, and I was like, well, maybe they're all dead. They didn't make it through the dead gate, so we looked at the live dead stain, and they're actually quite viable. So these ones over here are the dead ones, and these are the live ones. If we look back one gate further, we see that in the singlet gate, she has a huge portion of these things that are over here, right? That they fall out of the singlet gate. So I asked her, I'm like, what are you guys looking at? Like, these are some big clumpy cancer cell maybe? She's like, no, these are 293Ts. Okay, sample prep. These things must be just all balled up, and you're never going to get a good yield if you can't separate them out into individual cells. So almost half of them are in clumps. They're excluded from the sorting. And furthermore, if you're actually trying to sort and break off droplets, having clumps go through there is going to give you a really uneven break off. So in two ways, even though it said she had 100,000 cells in her tube, my guess is she actually probably had far less than that. So she's getting a really low recovery when she should be up in the 80% plus range. So this is a super important point. All right. So the last thing I want to talk about before we look at compensation, because I know everybody just can't wait to get to the compensation, is the one other thing you want to do before you start throwing things into clustering programs is to do a file cleanup. And what I mean by that is, we're talking about fluidics cells all lined up with lasers. So we want to remove any irregularities. So you can see that if we're going to use a time plot. So the easiest thing to do is take a parameter, plot it versus time. And you can see that at the beginning, we actually have a lot more cells coming through at this. So we're probably going to go ahead and just gate once the stream has stabilized. In this particular plot, if we're looking at a fluorescence parameter, it doesn't really matter which one, versus time, you can see somewhere in the middle, we had a clot, then we had a boost. We don't want any of this section to be in our data, so you're going to want to manually time gate that. There are definitely algorithms out there for automated data cleanup. If you imagine that you're doing a 40 parameter assay and you have 96 wells, it's going to take a lot of time to go through and manually do this. So check these out: Flow AI, Flow Clean, this QC program, as well as this is using a peak method and flow cut. They use things such as the skewedness of these individual parameters, whether there's outliers, does the mean change over time, et cetera. So if you imagine if your flow rate is changing, your overall MFI is probably going to go up or down. And within these 4 programs, they have various levels of automation to do this sort of gating for you. All right, here's your favorite part: evaluation of compensation or spectral unmixing, whichever one you're doing. How do you determine if your compensation is good or bad? I think that if these are the two samples, I think that everybody in the room would very clearly say that, well, this one seems to make a lot more sense in terms of having populations that are reasonably orthogonal versus this one here, which has a very large population that is very far below 0, so can't possibly be correct. How do we do that in a more systematic way? Will it affect my data if my single color controls are not good? I think that's obvious at this point that absolutely yes, it will. Maybe this is what we're really all waiting for. How the hell do I fix it? I can't just throw away those 40 mice and those 96 samples, right? First of all, using spectral controls, and again, this is some data that Meredith put together for us. If you use good controls that really reflect what's happening on the cells themselves, the unmixing process changes dramatically. So what is it that she did to actually make these populations go from something which is clearly overcompensated in this direction and this to something that is properly compensated? Although I would say that this sample here looks like it still needs a whole lot of data cleanup outside of this. So again, neither beads or cells are the perfect answer. So we already talked about all the work you're going into sample prep, all the work that you're going to be putting into your controls, all that work that you're going to be putting into your panel design. With a new panel, it is extremely useful to have both single colors and beads. And the reason for this is, yes, the beads are easier, so I will always unmix with them first. But the single cell controls can be used to see which dye did not unmix properly if you see problems like this one up here on the right. So which one of the dyes was the biggest problem here? If I have a single color control, I can quickly see how each individual color, when it's by itself, was unmixed without worrying about the effects of all the other fluorochromes. If I have single color cell controls and I made a bead matrix and I find I have a dye that's a problem, I'm going to take that cell control and I'm going to just swap it right into the matrix, pull that bead out of there. And honestly, out of a 40 color experiment, we found that maybe there were 3 or 4 that we actually had to swap out for the cell controls. So it's worthwhile to do that. And we will just know for our final experiment moving forward that we're going to use 5 cell controls, 35 bead controls. And as I mentioned earlier, it can help determine the loss of resolution in the full panel. So how do you look at these? Now, this happens to be FlowJo. I don't care what software you work in. Most of them have a way of looking at an n by n plot. Does anybody use these regularly? What do they mean? What is an n by n plot? Yell it out. It's one fluorochrome by all the other fluorochromes, right? So you want to see each one mapped against itself. This has nothing to do with what's biologically relevant. We're just seeing whether or not the unmixing worked. Just looking across here, we have negatives. We have things that are kind of centered around 0. We should not have things that are negative. So this looks wrong. This looks wrong. These are probably double positive. Remember, this is a fully stained sample. This one looks suspicious because as it gets brighter, it hooks out. This one is below the axis again, et cetera, et cetera. You kind of get the idea. Okay, so this was my fully stained sample, unmixed 100% with beads. I'm going to focus in on this one. This one looks like it's got the worst error. So I'm going to go to the single color control, and I'm going to ask, did the beads unmix? So these are now the beads in that one color, and you can see that these are unmixed correctly. So now I'm going to look at the single color cell control, and sure enough, this one is only positive for one single color, and you can see that it hooks quite badly. So this is, again, cells with a bead matrix. So now I'm going to take this one control, this one cell control. I'm going to flip it into the matrix, substitute it in. Your software might be different, but basically right here, I said, okay, instead of beads, I'm going to use my spleen cells. Okay? I'm going to use those as the positive and the negative. And you can see that that corrects the problem. So just swapping out one, I think this is a 22 color experiment, can very quickly fix what looks like a big hot mess. So this is definitely well worth it to do it. And you cannot throw this into gating or into your clustering program until all of these things are fixed first. All right, so this is the final result. This was one color. Then you're going to go back and you're going to review those n by n plots again, and you're going to do this iteratively until you have fixed all of the problems. And again, I'm not going to go through all of this for you. It was 22 colors. You don't really care which ones didn't work in my experiment. Once these are all orthogonal to the cell negative control, then you can move on with the rest of the steps. Okay? And those steps, as we mentioned before, are now going to be, they're properly compensated, they're properly unmixed. what is the resolution? Did my panel design do well? These are my single color, my fully stained. Again, this is another one. This is 27 colors in progress. This is 40 colors fully optimized, where yes, I have spillover spreading, but I can still fully resolve the positives. All right, so that's pretty much everything. We have this workflow that we went through today, from our scientific question and looking at our assays all the way down to sample prep, panel design, compensation, and unmixing. And then we're going to do this final data quality evaluation so that you can all get this at the end of the day. I think it's been a long day. He's got a lot of really great information, but I think we're going to have what, 20 minutes or so for a roundtable discussion. That's all right. So, yep. So if we're going to go ahead and join over here. Anybody has questions?
Now, yeah, will do. So the question was, what do we think about algorithms that allow you to do unmixing or compensation without any controls? Can you clarify if there's a specific one or like a specific provider that you've done that with or seen about that? Okay, got it. I was asking out of curiosity, not as a BD person. I think that yeah, so there are a couple of papers. There's math for doing it. There's a topic called blind source separation or unsupervised unmixing. So essentially, yeah, it's where you try to infer the spectra from the fully stained sample. It's mathematically possible. There's a technique called non-negative matrix factorization. There's a paper showing using that on Cytec Aurora data from a few years ago. The thing is, so I mentioned this concept of bias and variance in my talk. That bias variance trade-off is always there for these algorithms. And the way that one of those unsupervised techniques balances bias and variance may be different and maybe suboptimal compared to the linear unmixing approaches that we talked about. So the way I kind of think about it conceptually is like you have to pay the piper somewhere. And so the penalties for some of those unsupervised techniques, unless they're very, very sophisticated in their understanding of the actual noise on the system, may be that you end up with higher spread. I don't know that for sure, but that is kind of mathematically where the trade-offs might come in. There may be applications where it's so much easier to do it that that's totally worth it. And I think people are looking into it. But yeah, that's my impression from a technical perspective. Yeah, so there are a few versions. There is a paper called Autospill that came out of from Carlos Roca, yeah, in the UK. They do an iterative approach that looks at your single color controls and then sort of tweaks things to get them looking right. That then requires single color controls. You could do that with a full sample. These other techniques, the non-negative matrix factorization, it might be iterative internally a little bit, but it's basically doing some math without just sort of blindly trying empirically to say, given this data, here's the spectra that could have explained it, essentially.
There is an online question. This probably refers to some specific company or something, but doesn't name them, but claims that some companies doesn't really emphasize the importance of controls, single stain controls being as bright or brighter than the experimental samples. Have you encountered this kind of claim, and what do you think about it?
Yeah, I know what they're talking about, and I'll be happy to hear your guys' thoughts on this as well. So it is within the realm of like algorithm possibility to come up with a way to correct for errors that may come from a dimmer control than your fully stained. So if somebody has figured that out and is implementing it, that's great. If it's the same unnamed provider that I'm thinking of, there's also a lot of tools for fixing unmixing errors manually in that same software. So take that as you will. But in principle, there may be a way to correct for it. But yeah, from first principles, you can't kind of make up the data that's missing from those controls. It could, but yeah, like at least from the math, unless you're doing something really sophisticated to fix it, it's probably best to have them as bright as. Yeah. So Virginia's question was, does the control need to be as bright or brighter, or does it actually need to match the brightness of the fully stained sample? And I think that it's okay if it's brighter. You just eventually pay that penalty if it goes off scale. But in principle, it's okay if it is brighter. And in some ways that could be safer because it's ensuring that you're going to see parts of the spectrum and kind of the shoulder regions or on cross laser excitation. But there may be other reasons from a workflow perspective that it's better to have them the same, but purely from the unmixing perspective, it's okay if they're a little bit brighter.
It is possible, though, if you want to estimate those low levels and you have a dimmer control, to overcome that by simply running much larger numbers on the control to kind of overcome that statistical uncertainty on the off channels that have dim signals.
Yeah, there may be some cases where that helps. In other cases, it could be even if you had 1,000 events, the average is still buried in the noise, and then it wouldn't come out. But yeah, there's probably some shoulder ranges where just getting more would eventually pull it out of the noise. That's a good point.
Yes.
There is a second question online asking about, is there a situation where unstained universal control, unstained cells would be better than take a negative population from the experimental sample lab?
Yeah, so I think some of this depends on, you're talking about beads where they can pick up a dim signal and whether or not that's going to be correct. For the most part, for straight up compensation, I think that does not create too many problems, because what you're going to do is you're going to subtract that background with a dim fluorescence from the background with a very bright fluorescence. And so you're going to kind of decrease that small amount of fluorescence gets subtracted out of there as well, but you're still going to get a correct calculation across the board. I don't know that is actually going to work quite as well for the spectral unmixing, but it
Perfectly
works.
valid.
Yeah, so I don't think if it's, dim versus bright, it's going to really make that much difference.
I have one thing to add on that I did wrong a lot, and that I think a lot of people end up doing wrong, is when it comes to the control for your viability stain. This gets really tricky because the negative cell for a viability stain is a live cell, but a live cell has different autofluorescence than a dead cell, usually higher autofluorescence depending. So in practice, that's actually a case where you want a separate unstained dead cell as the negative control for your viability stain. Just spiking in like 50% live cells or using half killed and gating on the live is going to mess up, certainly for spectral, especially because if you're using one of these sort of UV 400 to 500-ish viability dyes, that is exactly where most of the autofluorescence signal is, and you can start to see some real unmixing errors. So that's a case where having a, not universal, but a separate unstained dead cell control is actually really helpful for nailing some of those subtle problems.
A slight change to that cells that you cooked the hell out of don't necessarily have the same autofluorescence as those that were just dead in your sample. So, and I have yet to bring me a sample that had no dead cells in it. So typically just not cheaping out on the unstained themselves. So running that unstained control and grabbing enough of the unstained control, you don't necessarily have to purposely kill them, but running enough of them so that that 5% dead or 10% dead is a big enough population to be your autofluorescence background. And then manufacturers would make it a lot easier if they would allow you to put multiple gates, small gates on those unstained, you know, one over the dead, one over the myeloid, one over the granulocytes, one over the lymphocytes, so that you could use, because that does make a massive difference. I actually had a slide that I pulled that really showed how just using a lymphocyte gate versus a myeloid gate versus everybody likes to draw the big all-inclusive gate really changes your unmixing quality, and you get errors, pretty substantial errors. You just can't simply do it like if I have my unstained, I can't draw one gate here, one gate here. What I have to do is create 3 tubes, save 1 tube, and then populate the other 3 tubes so that I can draw 3 different grades. It's an easy, every manufacturer has a workaround. It's just that they know that this is a problem, and we're starting to see the industry really kind of step up and say, yes, we're gonna fix this. And so yes, every iteration, I think the software gets better.
So I have more of like a why question that's been bothering me. I don't know if you guys have any feedback, but when we're looking at bead controls, single stain controls, I think we all agree to use the in-tube negative as the background, like we were talking about. But I noticed like some fluorochromes have different levels of that background. And I wonder, is that a artifact of the beads, using beads and the negative, or is it a biologically relevant something with the fluorochrome that leads to that negative background moving? I don't know.
So I've seen both situations. One is that if you're in the violets, the beads themselves are super autofluorescent in the violet and the UV regions. So sometimes that high background is not necessarily that it has more dye stuck to it, but that the actual autofluorescence of the bead is higher. If you compare it to the unstained in a separate tube and it's still higher, you're right. I have definitely seen some of the brands of beads that I ended up with some BV stained beads that had no negative population. So they were non-specifically sticking to the other bead. So that definitely happens. And again, so long as you have reasonable separation between the positives and the negatives, that kind of background building up on that is really not much of an issue.
I have a question that I would like addressed for some of the users. I still have a lot of users that bring me CD4 stained cells for every single control instead of their actual antibody. So I know you guys have touched on it, but if you could just again touch on why just using CD4 for every color is not the best control ever created.
Sure. So, I mean, it's a lot to lot issue, right? CD4 is a great antigen and it's available in every color. On the other hand, PE-Cy7 is available from lots of suppliers in lots of lots, and it's never the same. So your tandems need to be matched to the cells that are in your experiment. And also, I mean, for, you know, monomer dyes like a BV421, it can be acceptable to use CD4, but if you run every experiment like that with every dye, you're going to get stuck, yeah.
There is a question online again about the antibody concentration. Do you have to use the same antibody concentration for single stain controls and experimental samples, or is the emission spectrum going to be independent of the fluorophore or antibody concentration?
It's not about the concentration, it's about the brightness, right? So we've titered on our cells. We may need to titer if we're using beads on the beads. The object with the beads is to make them look like the cells, and the object with the cells is to fit them into the linear range of the instrument.
I have a question. Yeah, please go ahead.
why people think that because, we say, do your copies, mixing these, treat them the same as cells, but that means it doesn't mean the concentration, and there's no relation.
Thank you. My question is about how to titrate antibodies or in general gauge something if you don't have good strong positive. If you are trying to look for some really rare epitope, 0.1%, how do you titrate your antibody or put the gate for this negative versus positive?
I mean, this is not a good answer to this question, but if it's difficult to titrate, you probably aren't going to be able to determine a good titer with any great accuracy. So I might just go with a sort of a standard working concentration, which is usually somewhere in the 0.25 microgram per 100 microliter. I don't know that you can get a better result.
Yeah, you could also keep bumping it up until you start to see the negatives start to spread. So, well, even just yeah, exactly. I mean, if you don't see any positives whatsoever, then I'm not sure what you're staining for. You need either a clone that works or, I mean, you should have some small population. So you're going to have to run a lot of cells so you see even a small population. If that population is dim, again, you're going to start to see those negatives start to spread out. And then eventually they'll start also walking up as they non-specifically stick.
Yeah, and I guess I would say that maybe with a titration on something very dim, you're going to go from a shoulder to a big messy peak, right?
There is a question about, let's say, first running single stain beads and noticing that there is problems and then swapping that for single stain cells instead in your matrix. And the question here is that won't your negative autofluorescence in your controls also change and affect your results or create problems? Can you comment on that?
Do you mean when you see the negative fluorescence?
Well, the question is not very clear, but basically they are asking that
if you
can basically the mismatch of negative and positive, I guess.
Yeah, so if you see these little hooks, so these hooks that are kind of going negative, so you have some positives that are proper and then they kind of hook off into the negative direction, those are just indicating that the calculations for that dye just didn't match the matrix that was generated. So if that matrix was generated on beads, you're going to want to try a cell control to see if you get a meta matrix. And if you don't fix that, then yeah, it can have very serious implications for your data. So imagine that your data hooks off this way and you're overcorrecting and subtracting out basically signal from there. And now you have something, the other dye in the other direction is something that maybe you had a signal on and you want to know what the intensity is of that other dye. That's not going to be at all calculated correctly either. So you really can't just say, oh, it's okay to leave it hanging over there in negative space if this is going to be an assay that you want to get some real valid data out of. They really do need to be fixed. And I don't mean fixed by going up into the matrix and just randomly assigning a different matrix value to it. That's not fixing.
I think another part of the question might be like, is it okay if your controls have different autofluorescence from each other or from your fully stained sample? And the answer is yes. So there's two senses where autofluorescence is important in unmixing or compensation and in the workflow. The first and the most important is that the autofluorescence of the positive and negative within a single color's control are the same. That's exactly what Lisa talked about. The second is if you are unmixing autofluorescence in your fully stained sample as an additional color, then the reference control for that autofluorescence needs to match the autofluorescence of the cells you're looking at in your fully stained sample. But those two considerations are totally independent of each other. It's okay to use all beads if they all work, and then just have an unstained cell control for your autofluorescence unmixing control, as long as you're gating on the correct kind of autofluorescence population for the cells that you'll be looking at.
There is one more question online asking why people are not using more of the, for viability dyes, more of the lysine or the arc beads. Is there some drawbacks that they are not as good as cell controls? Why publications are not full of using these beads nowadays?
So the arc beads they're remarkably like convenient for sure, but they are also very bright in the UV and violet on their own, just based on the polymer that's used in those beads. So they give you pretty weird results in a comp or an unmixed matrix. It is tricky. I think Peter was saying a minute ago, it's difficult to get a negative control live dead that's dead, you know, and that's a good match. But you're much better off, I think, with cells in anything that's a reasonably complex experiment. I might use beads if I'm working in the 10 color space because it probably will work fine. When I get to a 40 color space, I probably am not going to want to use beads. And in particular, I don't know, on the Cytec spectral instrument anyway, it's problematic to use the arc beads.
It seems that, one more question, good. We still have a little bit time.
Yes.
Honestly, it's a great control to do that. So this is fairly software specific. So if you haven't used it, what they do is they show you. So all of the plots that we looked at today, you know, where they're showing you the intensity across all those channels. I think Karen showed a bunch of those. You may have showed a few. What they're showing you are the actual what's seen on the detector. So that is autofluorescence plus the dye. And in the Aurora, there's that, if you hit that next button, it will show you the plot where it's background subtracted. So in other words, they take whatever you said was the autofluorescence control, whether it was a negative bead or a negative cell, and it will subtract it out. And in all cases, you know, your dye signature, you know, typically they're in one region, right? They're not all the way across. So you'll have like a 100 nanometer space where you have a signal, say at 670 plus or minus 50, where all of those channels will have dye signal. And when you subtract that out, all of the ones around 400 shouldn't have any signal. There's no fluorescence out there. So if the autofluorescence was correctly subtracted out, you'll see that nicely go to background on the channels where you shouldn't see any dye signal. So it's a really great control, and you can do that on the cells as well. And if you're using the right cells, the dead cells for the dead dye, the lymphocytes for the CD3 dye, you should come down to background all the time very tightly. It's a really good way to evaluate whether that works.
Okay, I will thank our panelists today and all the speakers of the day. It has been a great day today, just the first one out of 3, so please come back tomorrow. We are starting with the breakfast again, 8 a.m. and the first talks 8:40 or thereabouts, the clinical session. Thank you so much.